Good morning and welcome to the second day of the 13th Annual Sentinel Initiative Public Workshop. I'm Mark McClellan. I'm the director of the Duke Margolis Center for Health Policy at Duke University, also an independent director on the boards of Johnson & Johnson, Cigna, Lyman Healthcare, and Prognom IQ. And I've received advisory fees for serving with Arsenal Partners, Blackstone Life Sciences, and MITRE. And I'm welcoming you on behalf of Duke Margolis for this meeting in collaboration with the FDA. We're convening this workshop under a cooperative agreement with the Food and Drug Administration to have a conversation. It's supported through the cooperative agreement, but this is not a federal advisory committee, will not be voting or making recommendations. The conference will be a success if we have a robust exchange of ideas about where we are and where we're headed on the many important dimensions of the Sentinel Initiative. During this workshop, leaders from the Sentinel Initiative, along with key collaborating partners, are going to share developments in their work over the past year and also look ahead to the future. So on the next slide, you can see that we began the summit yesterday, the event yesterday, with a keynote presentation from Dr. Patrizia Cavazzoni. Then we heard some high-level updates from Sentinel leadership in that opening fireside chat. That was followed by a couple of in-depth panels from the Sentinel coordinating centers to discuss implementation of their master plans along with other developments in their work. We heard about the latest efforts to improve data validity, methods for causal inference, and from all of this, generating effective real-world evidence for the fulfilling the Sentinel mission. Then we heard from Rob Califf, who joined us for a closing fireside chat. So you put all this together, there were a number of key themes and ideas emerging from Sentinel's work. One is that this program continues to expand, covering a broader range of data sets and timely questions and reaching more and more diverse populations throughout all of FDA, with the BEST initiative program extensions in the Biologics Center and the NEST initiative extensions and expansions in the Center for Devices and Radiologic Health. As part of that, new sources of claims data, expanded use of Medicaid as well as Medicare claims are broadening the patient populations represented in Sentinel's work. Also, new sources of data and collaborations involving not only claims, but clinical data for non-government covered populations. And we heard how these expansions have been critical in studying populations of interest for pandemic response. For example, in developing vaccine safety surveillance protocols, especially around populations like children and maternal patients, and how these new data sources are potentially informing Sentinel's work beyond the pandemic as well. Still a work in progress with more potential data linkage opportunities in process, for example, with data from immunization registries, but a tremendous amount of work, more of which I think we're going to hear about today. We also discussed the emerging use of electronic health record data, big development in recent years and months at the Sentinel Initiative, supplementing the traditional use of claims data. And we also heard about some of the innovative research methods and data analytic tools that have been developed to understand and make use of these new data sources more reliably. We also honed in on some of the work by the Sentinel Innovation Center and others to build out causal inference capabilities using real-world evidence. As Rob Califf mentioned at the end, potential for not only finding unknown safety issues or concerns, side effects in drugs and other medical products as they're used in real world practice, but also unknown and better defined potential benefits in these products as well. For that, you do need reliable not only data and methods, but full support to get to reliable causal inference. So lots of opportunities to continue to build on this. We heard about opportunities for relying on advanced machine learning and AI tools, but as Rob said and others said during the day, important to keep a hands-on perspective too, given the complex issues involved in the data. and the continuing to evolve data science around conducting these studies effectively and with confidence. And finally, we also discuss new collaborations between U.S. government agencies and international organizations. There is global interest in relying on this potentially large and growing base for developing evidence, but also global interest in making sure we're doing it right from the standpoint of data validity, appropriate fit for purpose methods, and effective execution, those key elements of the FDA's real world evidence framework. So all of these are topics that I think we'll have an opportunity to build on in today's discussion with a number of very interesting panelists and panelist perspectives in the topics that we're covering today. And that's on the next slide. Today, we're going to be more focused on Sentinel and BEST's work to support pandemic response, which of course you heard about yesterday and you all know has been a big focus for all of us over the past year and a half. We're going to begin by hearing about collaborations within the Sentinel initiative, including some unique international collaborations driven by the dynamic nature of the global pandemic. Then we'll have two sessions centered on BEST work to combat COVID-19. The first focusing on partnerships with other federal agencies to support a robust whole of government response to the pandemic. And the second will cover BEST work with collaborating partners to leverage near real-time data for pandemic response and the development of vaccine safety monitoring systems and protocols. So looking forward to all of these, and just a couple of other preliminaries before we get started. Next slide, just a reminder that Duke Margolis is part of Duke University, where we have a strong respect and honor the tradition of academic independence on behalf of our faculty, scholars, and all of our collaborators. None of us take partisan positions as part of this work, but our individual members are free to speak their minds and express their opinions on important issues. I hope you all will do so as well to make this as frank and meaningful a discussion as possible. And then on the next slide, just a few reminders about our virtual environment. We want to encourage everyone to participate, like many of you did yesterday, through the Zoom Q&A function. That helps us work in questions into the discussions that take place. And in some cases, our panelists were able to provide written responses to questions that we didn't have time to get to in the panel discussion. So please keep those Q&A questions and comments coming. Again, all of our audience members are encouraged to also keep the conversation going on Twitter. We have a hashtag Sentinel Initiative, hashtag Sentinel Initiative, all one word for that Twitter dialogue. And let's get started. It's my pleasure now to introduce our Center's Director of Real World Evidence Programs, Rachel Hendricks-Srupp, who joined us for some of the moderation yesterday. And Rachel, glad that you're back for more today.
Much and welcome back everyone, for those of you who attended yesterday and for those of you attending for the first time today, welcome. It's my pleasure to introduce our next panel. Our next panel will talk or at least focus on Sentinel collaborations for COVID-19 response, and our esteemed panelists for this session include folks from Sentinel's collaborations for COVID-19 response, and our Michael Blum, Deputy Director of CDER's Office of Pharmacovigilance and Epidemiology at FDA, Noel Kakoris, Research Scientist at Harvard Pilgrim Healthcare Institute and Lead Epidemiologist for the Sentinel System Operations Center, also Susan Winkler, CEO of the Reagan-Udall Foundation for the FDA, and lastly, Catherine Cohey, pharmacoepidemiologist on the Data Analytics and Methods Task Force at the European Medicines Agency, or EMA. So I'm happy to have Michael kick this off. Michael, take it away.
Okay, thank you. Good morning. I want to thank the organizers for the opportunity to join the panel. What I plan to do is briefly discuss the niche that regulatory agency studies fill and the value of the Sentinel COVID-19 studies in filling that niche. You'll hear more detail on those studies in the talks to follow. So if I can have the next slide, please. So why would a regulatory agency conduct a post-approval, or in the case of COVID-19, a post-emergency use authorization study? Well, there are a number of reasons that I show here. I'm going to focus on those that are most relevant to COVID-19 studies. And so when I think of Sentinel, my eye is drawn to the fifth bullet point here. Data sources available to the agency may be superior to those available to industry or academia. Certainly the size and the diversity of the Sentinel data sources make it a valuable source of data during a pandemic that is disproportionately affecting different populations in different geographic areas. The first bullet point here, regulatory agency questions may need rapid answers, is certainly relevant for a pandemic. Rapid answers are clearly needed to make rapid decisions, but the data need to be of sufficient quality so that the answers are not misleading. And so it comes back to the age-old question of balancing quality with timeliness. Now, you'll hear more about the rapid COVID-19 Sentinel distributed database that's been developed during the pandemic. You'll hear more about that in Noel Kakoris's presentation. And that rapid database was developed for this purpose and has been used to assess outpatient corticosteroid use, the natural history of coagulopathy, and COVID-19 in pregnancy. The second bullet point here, some questions apply to classes of medicines. I already mentioned the example of a study of outpatient corticosteroid use, which looked at both oral and injectable corticosteroids and would not have been applicable to any one sponsor. There have also been utilization studies of a large number of inpatient critical drugs used to treat COVID-19 or its complications, and the results have been informative in assessing potential drug shortages. The need to develop evaluation methods is really an important reason for regulatory agencies to conduct studies. And you've already heard mentioned several times at this meeting the difficulty with capturing oxygen-related therapy in hospitalized COVID-19 patients. That's a particular challenge, again, that Noel will explain in detail, but it required a broadening of data sources, and in this case, to include nursing documentation. So we are continually learning new methods for data capture and analysis through the Sentinel COVID-19 studies. Next slide, please. So on the topic of collaborative regulatory agency studies, I show here the opportunities and the challenges. What I've learned through the pandemic experience is that it's critically important for regulatory agencies to share information in real time in order to address an issue that can only be effectively dealt with on a global level. The pandemic's provided an opportunity for regulatory agencies to work together on a variety of COVID-19 studies, including the natural history of coagulopathy and COVID-19 in pregnancy. I don't want to belittle the challenges that are listed here because they are considerable, but I have a good deal of optimism that working through these challenges now will pave the way to even more collaborative regulatory agency research in the future. As you'll hear about shortly, the Sentinel work on COVID-19 and the collaborative work through the Reagan-Udall Foundation and between regulatory agencies has required substantial effort, but has resulted in significant advances. And my hat is off to my colleagues on this panel, as well as everyone who has worked arduously in this area to improve the public health. So with that, I'll turn this over to our next presenter. Thank you.
Thank you so much, Michael. Lots of things to think about here, and we appreciate you sharing your thoughts. I'll go ahead and turn it over to our next speaker, Noel Kakoris. Noel, I'll let you take it from here.
Good morning. Thanks, Rachel. Good morning, everyone. My name is Noel Kakoris. I'm the lead epidemiologist with the Sentinel Operations Center at Harvard Pilgrim Healthcare Institute. And over the next few minutes, I'll give you a high-level overview of some of the key work that's been done within Sentinel in response to the pandemic so far. Needless to say, I'm covering work done by many, many people within the Operations Center at FDA, within our various data partners, and also our scientific collaborators. Next slide, please. Just want to note that some of the work discussed this morning was supported in part by Hca Healthcare, and the views are my own and not necessarily those of Hca. Next slide. So for COVID-19 response, CDER is needed and will continue to need a variety of data sources that are well characterized, quality checked, reliable, robust, basically data sources that can cover their needs in multiple care settings, inpatient and ambulatory, that have the data elements of interest. And I'll talk a little bit about how Sentinel has been responsive to those needs. Importantly, years of long-term collaborations has enabled Sentinel to be really responsive to the pandemic by enhancing what we can do within the network, as well as bringing in more partners. And so what I'll do is summarize some of that work. And I also want to emphasize, as Mike mentioned very briefly, a couple of the projects that were facilitated by the use of master protocols. And really the collaboration was, as Mike said, lots of challenges, but also lots of opportunity. And I think the use of master protocols for some of this work really highlights some ways to address these needs. And I think one other thing to note is that for some of these protocols, as I'll talk about briefly, is that these are studies that were aimed at understanding the natural history of COVID-19 to some degree, and that work will be really important to inform future studies around treatment safety and effectiveness as well. So if you go to the next slide, please. On the Sentinel website, you'll find a lot of information about the projects that are underway or that have been completed. So I'm not going to be able to go into all the detail, but please go there. And if you hit the next slide, you'll see a reference to a manuscript that we published. Many co-authors and colleagues were involved in this that summarized some of the work to date as of a few months ago this year in response to the pandemic. So on the next slide, you'll see a very simple depiction of some of the data that's currently available within Sentinel for CDER. So on the left-hand side, it's sort of our typical claims-based network, the Sentinel distributed database, primarily commercial claims with some electronic health record information. And that work, which I'll describe briefly in a moment, has been expanded to include what we call the rapid COVID-19 database. The other three boxes are representing our EHR-based partners. We have PCORnet and their COVID-19 database. We have the data from Trinetics, and then we also have Hca Healthcare, which I'll talk about a little bit as well. So if you go to the next slide, this is the very high-level overview of the rapid COVID-19 Sentinel distributed database. So this was developed specifically in response to the pandemic. We have 6 of our typical Sentinel data partners, 2 large national health plans, which are Humana and Aetna, and 4 of our integrated delivery system partners. And as of very recently, there are more than 77 million patient IDs in this database, with more than 8.5 million SARS-CoV-2 diagnostic laboratory records. And what's in this database is all patients who were enrolled at these partners from early 2020 forward, regardless of COVID-19 status. And importantly, these data are updated much more frequently, and the data are much more fresher than our typical Sentinel data. By the end of this year, we're anticipating there'll have been 35 refreshes across the different partners, making this a really powerful database. And Mike briefly mentioned the uses to date so far. In addition to what he mentioned, we've also used this database to assess monoclonal antibody use, and we'll be using it to examine racial and ethnic differences in COVID-19 testing, hospitalization, and mortality. So if you go to the next slide, I'm going to show you a couple of very quick slides with a little bit of data, just a taste from Hca Healthcare. So Dr. Rich Platt yesterday talked a little bit about Hca for those who attended. It's a large hospital network in the United States. We are using within the Sentinel system data from 140 hospitals. These data are updated every few weeks. And FDA has been using it to study a variety of different aspects of the pandemic, some one-off analyses, as well as some long-term monitoring projects. So this is a slide from one of those longer-term projects, monitoring dozens of critical drugs of interest to CDER. So it includes medications that have been used for treatment of COVID-19, as well as medications that might be affected by supply chain issues as a result of the pandemic. So this is a subset of a few steroids. You can see their use over time in relation to COVID-19 hospitalizations. And this is just one bit of data that's been provided to FDA on a regular basis to help monitor what's going on in the country. If you go to the next slide, this is a little bit of data from most recent analyses conducted with HCA Healthcare data, assessing pediatric hospitalizations in the context of COVID-19. So these are data from mid-February through early August. The top graph is the number of hospitalizations with a COVID-19 diagnosis among children under 18. There are more than 1,600 at the time that these data are complete through. Gives you a sense of the pandemic among that population. And then importantly, the bottom graph is a little bit on the number of admissions where children have been diagnosed with multi-system inflammatory syndrome. And there are more than 300 cases as of when these data were run. So this gives you a sense of what's possible in this system. On the next slide, this has been, Mike just mentioned this, and then yesterday, Drs. Delpan and Platt also mentioned this. And I think it's important, we're mentioning this many times because it's really important, not just for Sentinel, but more broadly to understand that oxygen-related therapy is captured in ways not typical to how we use data in much of pharmacoepidemiology. And what we found here is that if you were to rely only on diagnosis and procedure codes within an EHR system in the inpatient setting, you would very much undercapture this indicator of severity. And so we wound up using nursing documentation, really gained a much more granular and comprehensive understanding of the use of oxygen in response to COVID-19. So I'm going to close this up with with just two quick slides mentioning the two master protocols. And I know that Susan Winkler will talk about the next one a bit. If you go to the next slide, this is a very brief summary of the natural history of coagulopathy and COVID-19. This work is being led by Dr. Vincent Loray at the University of Pennsylvania and myself. And this is a protocol that we developed and is being implemented both by EMA as well as Health Canada via C-nodes. And we're looking to understand arterial and venous thrombotic events among people with COVID-19 in general, and then also compared to seasonal influenza. And the master protocol has been really important here. And then also, as you'll hear from Susan, this has been a big part of some of the work within the RUF. And then lastly, there's been work, as Mike mentioned, to understand COVID-19 and pregnancy. So if you go to the next slide, this is a protocol that was funded by EMA called Consign. It's also being implemented within Sentinel. And this is aimed at understanding COVID-19 among pregnant women in terms of medications that are being used, and then also severity and clinical outcomes. So I'll stop there and thank you very much and look forward to the panel discussion.
Wonderful. Thank you so much, Noel. You presented a lot of great information about, some of the limitations to relying on specific coding mechanisms that run the risk of, or at least lead to the risk of undercapturing specific cases. So thank you so much for highlighting that amongst a lot of other useful information and points. I will go ahead and move on to our next panelist, Susan Winkler. Susan, I'll let you take it from here.
Great, thanks so much. And thanks for the invitation and the opportunity to join Dr. Blum and Dr. Kakoris and all of the speakers on the panel. I'm going to talk a little bit about the COVID-19 Evidence Accelerator, and particularly the champion role that the Sentinel Initiative has played in supporting this community and some of the research that they have done. So to get us started, I want to give a little bit of background on the COVID-19 Evidence Accelerator, so that we can better understand the really essential role that the Sentinel team has played in the accelerator. So if we go to the next slide. The COVID-19 Evidence Accelerator was a specific effort that was pursued by, is still being pursued by the Foundation and Friends of Cancer Research at FDA's request to bring together a community of data and analytic partners who were ready to dig into questions about COVID-19. So we convened it at FDA's request, and the financial support for the Evidence Accelerator from April 2020 to date, as we continue our efforts, is provided by both the agency and then some specific support from the Rockefeller Foundation for our work in a component of the accelerator. So if we go to the next slide, I noted that our primary partner here was Friends of Cancer Research. And so this is just a bit of background about why FDA would ask the Reagan-Udall Foundation and Friends of Cancer Research to convene the accelerator. And it's really based on prior work that we had been engaged in, not from an infectious disease standpoint, but from the real world data and real world evidence component. So we're happy to work with Friends on this initiative. If we go to the next slide. So some of this we've talked about a lot, but it's good to underscore, why did we need to look at real world data in COVID-19? One of the ways we talk about it in the Evidence Accelerator is that we were swimming in it, and we still are swimming in it. And learning as we go along, we certainly know much more now today in November of 2021 than we did in November of 2020 or in April of 2020 or earlier. But we're learning more about what all that is happening, and then that can be used, as many speakers have addressed, but to underscore, to prioritize research questions, to help FDA and others have a better understanding of what's happening with the evolution of the virus, and what are we learning, what can we respond to. From an FDA perspective, it's helpful to be able to see in some analyses near real-time performance of diagnostics or other tests that are authorized under an emergency use authorization, and near real-time vaccine performance authorized under EUA. So I don't think anyone in this group needs to be sold on the value or the importance of using real-world data, but certainly essential in helping to respond to a pandemic. If we go to the next slide. So just a bit of a snapshot. One of the things we do in the Evidence Accelerator is provide that environment for people to talk about and share what they're learning in real world data. And so these are just two of the snapshots that describe presentations we've talked about in the Evidence Accelerator that then helps the community understand what was happening. Some really, really interesting insights that the National Football League gained in their testing protocols, seeing what worked and what didn't, as far as distancing and masking, as well as the performance of the tests that they used. And then similarly in vaccine performance, an analysis in the Veterans Health Administration to say what was the kind of durability and the durability of protection against COVID-19 infection among vaccinated individuals within the VA. Sharing that information helped all in the community continue to learn about what's happening, not only with the evolution of the illness, but with the various interventions that we can take. Next slide. So this is where we get to part of where the Sentinel initiative has been such an essential partner in what we've done in the Evidence Accelerator. And in particular, if we talk about the work that we did here as a common protocol to repeat analyses on different data sets. And so I'm going to talk about what we call it the Parallel Analysis Project. And as Noel had mentioned, one of the projects that we pursued in that parallel analysis approach was the natural history of coagulopathy. If we go to the next slide. This just tracks where we are within the Evidence Accelerator, and that we're doing work in therapeutics, diagnostics, and vaccine, and that we'll be thinking about our next parallel analysis work. Next slide. So this is a description of a typical parallel analysis approach that we've been pursuing within the Evidence Accelerator, where you have a common approach. Data source number 1 does their work, data source number 2 does their work, data source number 3 does their work, and then we present those overall in a parallel approach. So we don't mix them together for a meta-analysis, but rather leave them separate for a parallel approach. In the parallel analysis, where we were able to collaborate so closely with Noel and Dr. Winkler and the Sentinel initiative, if we go to the next slide, was where they played, and click it one more time. In the natural history of coagulopathy, we articulated a key player or a lead player. So if you hit one more for the animation, the Sentinel team was, I think, acknowledged by all partners as a more than equal player. They were really the lead of this activity in designing the protocol and sharing that information, and then interacting and sharing with the other data sources. And I think it's fair to say that one of the things, certainly the project benefited from the Sentinel Initiative's leadership, but I think also it was helpful to the Sentinel team to have some of those conversations about what is it that we need to know and learn, what are the questions that are raised when someone else tries to run the protocol, and what's helpful in that collaborative approach. So within the natural history of coagulopathy, we have not only now the Sentinel analysis, but an analysis from partners at SciAps and from DataVan, from a few others and Health Pals, 3 different data sources who run the same approach. And then we're able to see, for example, well, how do the results in this natural history of coagulopathy look different in SciAps, which is a data set primarily of patients, well, all patients, oncology patients being treated for cancer. And so it's a different set, and we see some different results. So if we go to the next slide, if you click on an animation and then go through. So some pieces that Sentinel has also helped expose, and we appreciate their contributions, is the community coming up with some of these learnings about working with COVID-19 data. And we took the time to capture what's here. And on the next slide, I'm not going to go through all of these, but we have them posted on the Evidence Accelerator website, because there's things that folks working in this data know, but we may not remember when we want to look at it in the future, or thinking about how it's evolving. So some important things when we're talking about COVID-19 and real world data is that, for example, there wasn't an ICD code for COVID-19 until April 1st of 2020. And so before that, there's data, but it's under miscellaneous codes. So there's components like this. If you go to the next slide, and then I'll close out. Similarly, there were some dynamics that had to do with data set limitations, consistent with what Noel was just talking about in the oxygen data, and being able to do that. There simply isn't, there are a number of questions that we just need to understand aren't easily addressed within the data sets, but then working collaboratively can figure out the solutions to them. So with that, I'll just note these are some of the learnings that we continue to gather within the Evidence Accelerator. And the final slide is how we continue to do our work. If you click one more forward, and that's the idea that we want to come together and collaborate. We're so appreciative to the Sentinel team for their leadership on the natural history of coagulopathy, as well as their ongoing contributions to the conversation, so that we can improve the ability of the research and the RWD community to evaluate key questions around COVID-19. And with that, I'll turn it back to you. Thanks so much, Rachel.
Thank you very much. Thank you to all of our panelists. This was, again, another great presentation. And so I will turn it over to our last panelist for this session, Catherine Cohey. Catherine, you're on.
Yes, thanks very much, Rachel, and thanks as well for the opportunity to speak today. So my name is Catherine Cohey. I'm a pharmacoepidemiologist in the Data Analytics and Methods Task Force at the EMA, and in the next few minutes, I would like to share with you the EU perspective and some high-level examples and lessons learned so far from using European data networks. The next slide is the usual disclaimer that this is mostly my views and they may not be attributed to the EMA. Next slide. So, oh, this looks a bit funny, but yeah, I think you can I can still see it correctly. So first, where did we start? I will speak specifically in terms of vaccine monitoring. So we focused very early on preparedness with the Access project, which leveraged the data sources in the VAC4EU consortium and generated background incidence rates for AESIs, as well as template protocols for vaccine safety and effectiveness studies. We then launched what we called at the time an early hypothesis generating study when the vaccination campaign started. And the study includes primary data collection in 8 countries and a parallel cohort monitoring in electronic health records to look at AESIs and also at COVID-19 diagnosis prior and post COVID-19 vaccination. This study also provided a framework for the generation of new background rates for the evaluation, for example, of the TTS and the myocarditis signals. It is now transitioning into a larger study, also including special populations in the primary data collection part, while the EHR component will provide a framework for rapid signal assessment using the conception common data models and other tools for the of the VAC4EU consortium. In addition, we can also procure etiological studies to address emerging safety concerns through our EMA framework contracts, where we can access approximately, I think, 35 EU data sources through different academic organizations, vendors, and large consortia. For example, we have now a study ongoing with Erasmus and Oxford universities on thromboembolic events, which uses the OMOP common data model. In the next slide, I will discuss a few examples of international studies which were initiated through collaboration with FDA, as well as with other regulatory agencies, and the previous speakers already touched to these studies. What has not been mentioned yet is the eCore project, which was launched in June 2020 to address the initial need for real-world evidence on COVID-19 drug use patterns, safety, and effectiveness. And this project established cohorts with appropriate comparators in a number of countries using a common protocol and/or established common data models depending on availability. And the feasibility of this collaboration was demonstrated with a proof of concept study on patterns of use and risks associated with systemic glucocorticoids, as well as disease outcomes in hospital and primary care using the OMOP CDM. And what this pilot showed is that despite the many databases included, there was not surprisingly still issues with small sample size for rare outcomes and some quite substantial heterogeneity across countries, which precluded conducting a meta-analysis. However, the pilot showed that eCore can be used as a resource to address public health questions and allow large association studies to be performed rapidly. You heard a few minutes before about the Consign project on pregnancy, which started in July 2020 and collects data on the impact of COVID-19 treatments in pregnancy, with the ultimate goal to guide decision-making on treatment options in pregnant women. You heard about the objectives before, so I will not repeat them. I will just say that the project uses 3 work packages, which use 3 data sources, conceptions for the EHR component, the Cohey network, which uses an online registry, and the international network of obstetric survey systems that collects data from hospitals. So the study is ongoing, and we received interim results on drug utilization during the summer. We are now scaling up to perform a meta-analysis with data from other sources. FDA colleagues are adapting the EHR protocol within Sentinel, and colleagues from Canada, the UK, Japan, and Saudi FDA may also contribute. We're expecting results at the end of 2022. And ultimately, this project should serve also as a global framework beyond COVID-19 to study medicines in pregnancy. I will, for the sake of time, I will skip giving too many details on the third example. You heard already about it, the study on the natural history of coagulopathy and use of antithrombotic agents. This was initiated by FDA, as you heard, we tested the feasibility of using this joint protocol. So in Europe, it was conducted using, again, the OMOP CDM. And what we did is we added on top of the original protocol a cohort of vaccinated subjects following the TTS signal a few months ago. So this is, we just got the result. We are just digesting the report. Next slide. So yesterday in the session on causal inference, my colleague Gian Mario Candore gave an overview of real-world evidence generation at the EMA and presented Darwin. So I will skip this and focus on the next slide on how Darwin, once set up and running, will serve as a central pillar for health crisis planning and response in Europe. So I think we all know by now that the informed response to the pandemic has brought an unprecedented interest in real-world evidence, as well as a lot of scrutiny. And we already have real-world evidence use cases from the current pandemic, and these use cases will support the role of Darwin in the response to future crises, with an operational infrastructure for conducting studies, which is part of the updated EMA mandate. So these the use cases, you can see those on the slides. They include monitoring the use of medicines to predict demand and shortages, conducting natural history studies to support the development of vaccines and therapeutics, generating evidence to support the repurposing of existing medicinal products, and monitoring the post-authorization safety and effectiveness of vaccines and therapeutics. Next slide. So what have we learned from COVID-19 multi-database studies in Europe? From a regulator perspectives, and this was highlighted before as well, we need timely answers and high quality evidence. And we need also to focus on strengthening all steps of evidence generation, from data collection to the ultimate appraisal of the data for regulatory decision making. So this has led to this unprecedented collaborative efforts, and we have seen that readiness is critical. Just mentioning as a good example, the definition of AESIs and the generation of background rates. We have seen that large healthcare databases from several member states can be used, and rapid analyses are possible, but there is still room for improvements in terms of linkage to hospital data, having vaccination data stratified by age and gender, having laboratory or even genomic data available and valid, working on case definition and coding for new events of concern. There's still the issue of heterogeneity of data sources, lag times for data availability, and the need for outcome ascertainment. So still lots of work to do, and hopefully Darwin will solve some of those remaining caveats. We have also seen the timely exchange of information, transparency and communication between all relevant stakeholders, including regulators, EU data partners, academia was key to share information, data, experience, and leverage this knowledge to develop a global public health strategy. I just would like to highlight here the importance of large data networks to evaluate effectiveness, because we've talked a lot about safety, and I want to take the opportunity of this panel to mention that as part of the revised mandate of the EMA and the European CDC, the two agencies are now working hand in hand to assess the feasibility of using EU data sources to generate vaccine effectiveness across Europe. And in my last slide, I will not go into all the details, but this is really part of my daily life. So I wanted to highlight that as the pandemic continues to unfold, we will still need a high level of agility and readiness to keep up with a very changing environment. And on this last slide, you can see some of the research questions that large data networks hopefully will be able to address as we continue fighting this pandemic, and here focusing on vaccines. So with that, I'll hand over to Rachel, and I'm very much looking forward to the panel discussion. Thank you.
Thank you so much, Catherine. And thank you again to all the panelists here today for this session. You know, we started off the conversation, you know, first and foremost, knowing that, you know, FDA has a need to conduct its own studies, especially studies that require rapid answers during public health emergencies like COVID-19, the current pandemic that we continue to be in. It was really interesting to learn about how FDA was able to collaborate with Sentinel and also EMA to tackle some really unclear questions, particularly around understanding the natural history of coagulopathy in COVID-19 patients. There were some data challenges, obviously, along the way, as we all heard, how ICD system codes are just not always in alignment. They weren't in alignment at all, in fact, until April 2020, when the codes became available for COVID-19. So that alone was a big challenge. And then also reconciling, those codes with codes that were used prior to that. That must have been a big challenge. And hearing that you all were able to come up with, or at least develop protocols to address some of those challenges, amongst others, but really important things or lessons learned rather for the future as we prepare to continue to tackle this current pandemic and future pandemics as well. So thank you again for sharing all of your insights, lessons learned, again, all of the steps that you took to get to where we are today. So we do have some discussion questions to kick off a broader discussion with everyone on our panel today. We have a couple of questions in our Q&A as well that we'll try to get through in the time that we have. But I will start off with, our first question here. What capabilities have we built or leveraged during the pandemic that can prepare us for implementing more accelerated responses to public health emergencies? And I know, Catherine, you talked a little bit about Darwin. And so, maybe you can say a little bit more about, Darwin, what you hope for that particular program. I'm particularly thinking about real world data and real world evidence. You mentioned that you still need data that's fit for purpose, that's high quality, that's timely. What are your thoughts moving forward, around how we can start to get there? Around having the data that's of sufficient quality and timeliness and so forth.
It's a very broad question, but I think what Darwin, the concept around Darwin really is to be agile and also evolve over time as we characterize more and more data sources. So the aim is to expand to more data sources along with the European health data space being established. We will also look at interoperability between data sources, and of course the network will need to adapt to new needs, to new technology, embrace new stakeholders, leverage expertise of national competent authorities in Europe as well, academia, and learning from other agencies and other initiatives such as Sentinel, Eden, Odyssey, etc. There's also a need, I think, to look at having, and this is one of the first objectives of Darwin, of having a metadata catalog. So the coordination center will work on this as soon as it's being set up. So approximately around next year. And I think during all the previous talks, there was a lot of focus on fit for purpose real world data. So there will be, again, I'm using the word harmonization. There will be an effort to harmonize in terms of data quality, data completeness, etc. But of course, it's a very, very large endeavor, and we are only getting started with the coordination center being set up next year.
Wonderful, thank you so much. And we do have a question that might be along that vein in our Q&A. So, thinking about opportunities for future partnerships across data systems and centers, thinking about the NIH-funded N3C, the industry-led COVID-19 research database, or even PCORNet as Noel, you talked a little bit about today as, being one of the EHR system sources or databases, that have whereas we have opportunities, to leverage these sources to learn more and do more work and prepare for future pandemics. So Noel, what are your thoughts then? And I'll open this up to everyone else on the panel. Are there thoughts around future opportunities to leverage other pre-existing or newly developed databases or data sources to continue this work? What are your thoughts there? Because I think there are lots of opportunities, in fact.
Certainly agree. I mean, I think that I think one of the biggest things is making sure we don't lose the momentum. So I think, as you've heard, all these agencies have done so much work to be responsive, and I think now it's about maintaining what's been developed and then trying to look ahead and see what comes next. And so I think that as we've learned so much from the collaborations and partnerships that we have already and the data sources we have, it's identifying what's missing and then going to find where we can identify the data that we need. So I think that it's also about doing that in a coordinated way because I think that's one of the challenges too. So yeah, I think that there's part of the success, I think, for Sentinel in particular was that there was such good groundwork done. And now I think it's about expanding on that and sort of perpetuating it.
And if I could, I think there's the opportunity, part of what we saw within the Evidence Accelerator was the community aspect. And for Sentinel to kind of come into the community and play a leadership role there. Then there were other questions where it didn't quite fit with Sentinel. So it was a different participant who took a leadership role, but you had, because the community existed, you could have the conversations and think, where is there the strength for Sentinel leadership? Where is there the strength for perhaps a different approach? But you could have those conversations and kind of work it out because the community was gathering and learning and sharing. And I hope we keep that, the gathering, learning, sharing continue in some component ongoing, because it will just make it easier for those collaborations to say, okay, this is something that we would want PCORnet to lead on, or a different partner.
Thank you very much for your comments. So Michael, in your presentation, you talked about one of the major challenges being data standardization, or perhaps the lack thereof in some cases. And we heard from our other panelists that for this particular collaboration that you all undertook, you were able to leverage a common data model that works for you, either leveraging it on its own or adapting it. Catherine, as you mentioned, adapting it for use and implementation within the EMA context. So thinking a bit more about opportunities there to refine and improve on a common data model. What do you see ahead?
Yes, so there have been advantages to using similar or the same common data models, particularly in the collaborations that we've undertaken with CNODES within Canada, where they use the Sentinel common data model. But to date, in terms of our collaborations with EMA, because it's a different common data model, we've generally used common protocols. But we're looking into, in the future, potentially doing meta-analysis of results across regions, which will be a very interesting activity. I think Noel, you could probably speak further about the specific harmonization of common data models across regions.
Sure. I mean, just I guess briefly, so the Sentinel common data model is sort of highly flexible, so that's one of its major benefits. But I think more to the point of what it enables is when we're all using the same common data model, it's the most efficient of the efficient approaches. So CNODES, for example, can take exactly the same program that we ran with some minor modifications and run it. And actually, it's happening outside the context of COVID-19 in the UK and Denmark as well. So those two countries have transformed some of their data into the Sentinel Common Data Model and are participating on a Sentinel-led effort to conduct a study there. So I think there's lots of opportunity outside of COVID-19. But then, as we've heard, you don't have to have the same common data model per se to do all this, to do collaborative work. And I think the coagulopathy work with EMA has been a really nice demonstration that you get people together with an intention to do something deliberately together and to make sure that we implement studies in similar ways, and then we can do a meta-analysis as a result. So even when it's not exactly the same, there's still plenty of ways to collaborate and really take advantage of all the different data sources.
And I like how you all sort of brought it to, you know, thinking about where we are today, what are opportunities, or where are there opportunities to do this kind of work or leverage these resources in other non-COVID-19 contexts. And Susan, you mentioned during your presentation that at the Reagan Udall Foundation, you all were able to draw on lessons learned and tools that you already had available from prior RWE and RWD studies, you know, from other cases. So what were some lessons learned there, you know, thinking toward the future, you know, what do you envision then as opportunities moving forward, perhaps as an extension of COVID-19 or outside of COVID-19 from an RWD, RWE standpoint?
So I think some of there's a couple of principles that kind of rise to the top, I was thinking about it as the mention of kind of a meta-analysis and putting all the things together. What's essential there is that you have the rigor and the transparency of who are, you know, what do we know about that data set, and what do we know about the approach that we're taking within it, and what have we kind of worked through in the different approaches. One of the things we see in the evidence accelerators, because it's different types of data holders who then have different components, you have to work through and say, we might not all get to we won't be able to align at all, but can we clearly articulate what's different? And the ability to do that and go through a disciplined process and rigorously and transparently say, here's the approach in data set 1, or here's the approach in data set 2, and then be able to then compare at the end and say, what do we see across all, or what do we see differently, because we have some of that rigor and transparency versus trying to, I guess it's one of those where I'd say it's all hands on deck, but all hands with a role versus all hands trying to do the same thing. So what's the strength and the role that one partner would play versus another, and therefore what do you get as the result from that? So if we could do that quickly, that actually could apply to a number of different projects where we could say, you don't all have to have the same skill sets, but are we playing to your strengths to find that role?
Yeah, and actually that's a great segue to a question that we have in the Q&A, whereas an attendee asked about, you know, how, or at least provided there some insight around potentially Sentinel's initiatives being duplicative in some ways. So, you know, you just talked about, you know, bringing everyone to the table with their skill sets, but, you know, distributing the skills and the skill sets and applying them, you know, across the board where it's needed to sort of optimize the outcome and the effort. So thinking about duplicative efforts, you know, too many cooks in the kitchen and tackling one problem, and rather, you know, focusing on how we can sort of distribute that talent and those skills appropriately to, again, address the issue effectively and rapidly. You know, how can we assess, or how has the industry, or how has everyone here even thought about assessing duplicative efforts and addressing them to make sure that we're moving forward and not, you know, in a hamster wheel, I guess. Any thoughts there?
I'm gonna guess everybody feels a little bit like being on the hamster wheel, and that we've been on there for a long time. So it's a really big hamster wheel, but I think we've gotten much better in just the communication about what's happening and who's doing what. And that's the first step to what is duplicative and what's not. And sometimes what on the surface appears duplicative, then you can kind of parse out and say, well, actually, we're looking for this component and divide that out. So I think there are, I would observe that I expected to see far more duplicative activity than I think we have. And that there's been a lot of sharing. There certainly has been some, but there's been a better kind of alignment and sharing and a level of candor about the strengths and weaknesses of various approaches and various partners. That's been helpful, and another piece we should keep.
And then along these lines, I think I would use the term rather than duplication, and I totally agree with what you just said, Susan. I would use the term complementarity, and we've seen this very much in the last few months when we had new signals for safety concern. We had no idea about they were unprecedented, such as TTS or the multi-inflammatory system syndrome. So this was pursued on the FDA side. It was pursued on our side. We was using different data sources. We also had different vaccines authorized in our different regions. So this, at the end of the day, it's all coming together to help us assessing a signal. So I think it's all very complementary what we've all been trying to achieve.
Wonderful, thank you. So I do see a couple of questions in the Q&A. So one attendee asked about the timeliness and completeness of mortality data of deaths that are occurring not within hospitals for COVID-19. So thinking about data sources, all the data sources that were leveraged here or that could be leveraged, how, again, thinking forward, is there or have there been any lessons learned around data strategies to better capture these sorts of challenging cases, whereas deaths are occurring outside of hospitals and thus are not necessarily captured in a timely way? Just thinking about real world context and also real world data living within EHR systems. How are we thinking, or how have all of you thought about challenges around those particular scenarios or scenarios like that? And then I know, again, some of you have already drawn on scenarios in which, the ICD codes are not necessarily appropriate or unclear, and then therefore drawing us any conclusions or assumptions about that data also, sort of puts us a little deeper in the weeds if we're not careful. So I open this up to the panel. What are your thoughts about that moving forward in terms of how we can start to do better or think about better ways to capture data in more timely ways?
I can start it off. I've spent a fair amount of time thinking about this. So I think specifically around mortality, it's just a really big challenge in this country to get timely mortality data outside of the hospital. There are some situations where on a small scale, there may be more fresh and updated data available, but it's not to sort of punt the question, but in some ways it's a national problem. And so I think it actually requires a fair amount of discussion on a high level, I think, in different agencies about how to really handle this. I think more broadly, so much of this comes down to making sure you really understand your data source, that you have access to the people who generate the data, curate the data, because I think a lot of this, you can have some assumptions that wind up being wrong if you're not careful. So we spent a lot of time, for example, really looking at the completeness of information in the hospital setting for the coagulopathy study, because we're looking at inpatient-based events. And so we had to really understand on a individual data partner level, the completeness of the information so that we could design our study accordingly. And so I think that is something that has to be done for all of your major data questions along the way. And it's no shortcutting if you're trying to do it quickly with as much data as possible, as robustly and appropriately as possible. So yeah, there's not an easy answer to it. It's just being careful and thoughtful.
Michael, you talked a little bit about, the FDA's priorities in, conducting rapid studies, especially, looking at therapeutics or treatments post-approval, and within that post-market context. So what thinking, I guess, or at least framing my prior question around that, what are your thoughts from the FDA standpoint?
Well, actually, I've been thinking about your discussion question having to do with Sentinel extending beyond its core mission of safety, and what we've developed during the pandemic in terms of extending beyond safety issues. And I think that in terms of near real-time data, it's really been useful in terms of more effectively monitoring drug use, which is really important for our ongoing safety surveillance, as well as for addressing really time-sensitive issues such as drug shortages. So in that area, I see that continuing to be very useful beyond the pandemic. We have not done effectiveness studies of COVID-19 therapeutics, but we've been laying the groundwork for the effectiveness studies. So for instance, the natural history studies was, Sentinel actually going beyond its core remit in terms of developing natural history protocols and doing that COVID natural history studies, and that lays the groundwork potentially for a future treatment impact study. We have also been exploring data sources for linking SARS-CoV-2 variant sequencing data to that link the sequencing data to the clinical data, so that we could potentially do effectiveness studies of therapeutics specifically against specific variants. That's a very challenging area, but I think it's an important area for the future. So I just wanted to mention those areas that we've been exploring beyond the core mission of Sentinel.
Yeah, thank you for that. Well, we've got just a few more minutes left for our session. So I want to ask everyone a final takeaway type of question. What would you say, and I want everyone to answer this if we can, what would you say is your top lesson learned from this collaboration during COVID-19?
Preparedness.
Say that again.
The first word that popped to my mind is preparedness. Working on preparedness very quickly, because it all evolved very quickly, and I think we were fairly prepared.
I would say do it fast, but do it well.
Yeah, too. Yeah.
And part of that is that candor and engagement. So what do you have? What do you have questions about? What can you work through together? And which will slow down some of the speed, right, Michael?
Right.
but you may end up in a better place.
And I think trust, I think, is the maybe the second word after preparedness. I think a base of trust within each system, within each network, and then across all of the different organizations. It's really the only way that any of this can happen in general, but then also in an emergency response kind of situation.
And would you say that the tools that have been built so far among your team, perhaps among others, are those tools able to get us there? Or what is your opinion, or how do you envision the tools that we've built so far getting us to where we want to be based on those lessons learned?
Yeah, so obviously I have a very biased view here. So my colleagues built most of these tools. So I think that, we had a really good groundwork to start from, years of investment and building and development of relationships and analytic capacity on every level. You can't do something like this in a minute, and you can't do it under duress. So I think that we had a really good groundwork to work from, and then it's really about how do we leverage this and keep moving it forward, because there's a lot more ahead, I think, for COVID-19, and then who knows what else is coming.
Well, thank you very much. Again, I can't thank you all enough for this wonderful panel today. You know, we talked a lot about you know, the work that you've done around understanding the natural history of coagulopathy and COVID-19, the infrastructure that's been built along the way, your perspectives and opinions about, you know, what's next, you know, what can we do better, what are some lessons learned. Trust came up at the very end. It also came up at the very beginning in Michael's presentation. So, you know, I think it's a good way to sort of, you know, end it there, or at least connect the dots there around, you know, trust in the systems, the infrastructure, the people that are working within the systems, and everything else. And I think we have a lot of opportunities ahead to steer toward creating better infrastructure to more rapidly and rapidly respond to future pandemics and other public health emergencies. And, you know, thanks to everyone, like those here on our panel, for doing that for us. So thank you again for your time. Thank you to the attendees for your questions. And I will turn it back to Mark. Thank you again.
All right, great. Really enjoyed the panel. Rachel, appreciate your moderation. So we've heard a lot about Sentinel activities related to natural history, special populations, looking at therapeutics and other issues. Now we're going to focus in on the activities of the Biologics Center, CBER's Biologics Effectiveness and Safety, or BEST system, and its major role in issues related to vaccine surveillance and understanding in a wide range of clinical settings, particularly in the United States. We're going to do this in two panels. First, hearing from BEST's COVID response team within government, so including the Biologics Center, including perspectives from the VA and other parts of the federal government. And then in the next session, we're going to hear from the BEST partners in the COVID-19 response. Again, a lot of focus here on the very important issues around vaccine safety, access, and other issues where the BEST system had to surge quickly in response to the pandemic. Our panelists include Weili Wong, who's Associate Director for Innovation and Development in CBER's Office of Biostatistics and Epidemiology at FDA, Eric Weintraub, who's Project Lead for the Vaccine Safety Data Link project at CDC, another critical partner in this federal effort. And then Fran Cunningham, Director of the Center for Medication Safety at the Department of Veterans Affairs Pharmacy Benefits Management Services. So we're going to hear some opening remarks and presentations from each of these three federal speakers, and then we're going to have some discussion. Please keep those Q&A questions and comments coming. Weili, I'd like to turn to you first.
Thank you very much. Good morning, everyone. Could I just thank you. Next slide, please. Thanks. On behalf of our team lead, Dr. Azadeh Shoaibi, and the army of persons behind the FDA Biologics and Effectiveness Safety BEST system, today I'd like to share with you what we've been doing the past 12 months since we last spoke in terms of vaccine safety, effectiveness, and expansion of the BEST infrastructure. The focus actually will be on how we actually coordinate, how we are coordinated together with our federal agencies for a unified pandemic response. Next slide, please. So when we last met in October, next slide, please. At that time, there were only phase 3 trials for vaccines, not yet any authorization. We have since then completed these studies shown here. One of these actually is a effectiveness study for therapeutics, and that is in a large EHR systems for convalescent plasma, where what I'm showing here actually is that, and the theme would be that BEST has a range of real-world data that offers flexibility for us to respond to the pandemic. So in this case, if you would like to do effectiveness study for therapeutics, in this case, convalescent plasma, we went to directly source one of the largest health systems in Texas, because we found that one of the ways we have to actually account for confounding by indication to compare between those treated and untreated, we actually have to account for oxygenation requirement. In this case, among other things, we used oxygen therapy flow sheets for mechanical ventilation. Next slide, please. Now, since then, with 3 vaccines authorized and 1 licensed, we have been working on actively monitoring vaccine safety and vaccine effectiveness. For this panel, I'll be focusing on vaccine safety. Next slide, please. That's because Dr. Rich Hca and our other best collaborators will cover the rest of the both safety and effectiveness in the subsequent panel. For this panel, I'm pleased that we'll be joined by our CDC colleague and our VA colleague, who work very closely together in unprecedented coordination of our large distinct surveillance systems, but yet a coordinated, unified front for pandemic response. I want to acknowledge that our colleague, Dr. Jeff Kelman from the Centers for Medicare and Medicaid Services is unable to join us today, but CMS has been one of our valuable federal agency partners. We've been working with CMS, FDA and CMS have been working together for the past 2 decades, particularly for vaccine safety, and none of this work, and all of this work actually has been very valuable together with our partnership with CMS. Next slide, please. So one of the things that I pointed out is that we have this range of real-world data here, claims, EHRs, linked claims, EHRs, and in terms, I mentioned to you how we used EHRs for effectiveness for therapeutics, because that is kind of the purpose that's needed for that. And then we also used linked claims EHRs for validations of outcomes. And in terms of pandemic response, we found that that's very helpful both in EHRs and linked claims EHRs to very quickly develop the algorithms and to validate that. In this case, for vaccine safety, there was the TTS, or the thrombosis in presence of thrombocytopenia, that required us to validate in EHRs looking for the imaging results. And next slide, please. One of the things that we have used for the main real-world data source that we use here actually is actually the large claims databases. And there are 2 things that we did. One is that we've got faster data access. For example, pre-registered claims. Second one is faster refresh. For example, either bi-weekly or monthly refreshes. We use that database for the large claims database for vaccine safety for this range of activities, in this case of signal detection, which is real-world surveillance or rapid cycle analysis, and then followed by, when we get signal there, followed by we characterize that in pre-specified steps, either through additional analysis, and among this also includes cross-checking with other federal systems, and then we verify that it's fully adjusted epidemiology studies. Next slide, please. And we coordinate all of these responses in regular meetings and also direct interactions. We have planned for all of this prior to authorization, and then after authorization, we conducted these studies in coordination. We share results, and we regularly actually share study designs and coordinate our studies. Next slide, please. And we give you examples of how we work together and what it is. On the right-hand side, you can see these are potential adverse events or special events here for our near real-time surveillance. And this, prior to authorization, we've actually discussed and coordinated with both CDC and with VA and our other federal partners. And on a regular basis, in this case, either bi-weekly or monthly for those under 65, and weekly in CMS for above 65, as data accrues, we test the hypothesis that the post-vaccination rate is higher or elevated versus the background rates, or in others, versus in absence of vaccines. And if there's any signals, this will be evaluated in robust epidemiological studies. Next slide, please. We then report these studies in FDA communication. So in CMS, for those age above 65 years old, we have found that there is a safety signal for pulmonary embolism, acute myocardial infarction, immune thrombocytopenia, and disseminated intravascular coagulation. We reported this, we had transparent communication. We then shared immediately, rapidly with our federal partners, and we continue with our characterization and also our conducted a fully adjusted study for signal verification, and that's about to be completed. Next slide, please. So another example is that when we also get signals from others, for example, international regulators or independent surveillance, we immediately then do some ad hoc analysis. In this case, this is myocarditis and pericarditis, that's inflammation of the heart. And for a specific age group for the mRNA vaccines, this is for those males 18 to 25. We provided real-world evidence for regulatory decision-making during advisory committees by providing both incidence rates, comparing incidence rates between the mRNA vaccines, and provided these as excess risk for this input. Next slide, please. And while doing that, as I mentioned that we had shorter data lag, we had more frequent data time refresh for pandemic response. We also continue to build the infrastructure for pandemic response. In this case, we augmented the capture of vaccination in claims database with, next slide, please, vaccine registries. And that is the immunization information systems. Here you see for our best sites, we have around 9 that's actively sharing right now in links, and then around 20 that's onboarding. Next slide, please. So in summary, we actively are monitoring the safety of COVID-19 vaccines, in particular in this unprecedented, coordinated federal interagency pandemic response. Next slide, please. And as we've mentioned, because most of these vaccines are under authorization, so therefore generation of real evidence to support our federal, to support our regulatory decision-making is pretty crucial. And we have been generating these actually support either via through FDA communication or through advisory committee, and one of the examples is to inform benefit risk. And in general, this is in general what we'll be doing for the next upcoming year is to respond to the pandemic response and to have the flexibility given our infrastructure here and to provide real-world evidence to support regulatory information, regulatory system. Next slide, please. I'd like to thank the army of persons behind this work, unable to state all of them here, and our federal partners, CDC and VA and CMS, whom you hear next. Thank you very much.
Great. Thanks very much, Wheelie. Covered a lot of ground in those remarks. Looking forward to some further discussion too. Next, I'd like to go to Eric for your opening comments.
Hi, can folks hear me? It's Eric Weintraub.
Yes, can hear you just fine.
All right, perfect, thank you. Yeah, my comments are just gonna be verbal, so start my video here so you guys can see me. Thank you so much. Hi, good morning. I'm Eric Weintraub from the Immunization Safety Office at the Centers for Disease Control and Prevention. Thank you for the opportunity to participate as a panelist at this current year's Sentinel Initiative workshop. COVID-19 vaccines are being administered under the most intensive vaccine safety monitoring effort in the history of the United States. The Immunization Safety Office at the Centers for Disease Control and Prevention has a long history of monitoring the safety of vaccines that are currently licensed and or under authorization under the EUAs for use in the United States, and then recommended by the Advisory Committee on Immunization Practices. So the CDC has strong complementary systems in place, both new and established. For active safety monitoring for COVID-19 vaccines, the CDC developed and implemented vSafe, a smartphone-based monitoring program for COVID-19 vaccine safety. This tool utilizes text messaging and web surveys to actively solicit participants for adverse events and establish routine health checks. vSafe was established to complement VAERS, the CDC's and FDA's spontaneous reporting systems. VAERS has many strengths and limitations, but it can rapidly detect safety signals for rare events and covers an extremely large population. In addition to VAERS and vSafe, the CDC has CISA, the Clinical Immunization Safety Assessment Project, in place. CISA supports U.S. healthcare providers and health departments on complex, often unexpected clinical vaccine safety events and questions. CISA also assists with evaluations of patients with adverse events after COVID-19 vaccine or in making clinical decisions about administering COVID-19 vaccines to a person who may be at risk for an increased event, for an adverse event. Lastly, to complement all of these systems I've mentioned in the Immunization Safety Office, we have the Vaccine Safety Data Link. The VSD is a collaboration between CDC and 9 integrated healthcare organizations and is predominantly a system that relies on electronic health records and excels at conducting chart reviews in a timely manner. The VSD has an extensive toolbox when it comes to conducting vaccine safety studies, but one of those important tools is rapid cycle analysis, known as RCA. The VSD has been conducting RCA of COVID-19 vaccines since early December 2020, and the VSD is using a concurrent statistical sequential method and is analyzing approximately 23 pre-specified outcomes that have been critical with safety assessments, outcomes such as myocarditis, GBS, anaphylaxis following COVID-19 vaccines. And then importantly, in order to review this wealth of safety data being analyzed in these systems, the CDC established a new working group, the COVID-19 Vaccine Safety Technical Work Group, known as VAST, V-A-S-T. And the objectives of VAST are to review and evaluate and interpret post-authorization or approval of COVID-19 vaccination safety data, serve as a central hub for technical subject matter expertise from federal agencies conducting post-authorization approval safety monitoring, advise on analyses, interpretation, and data presentations, and then provide updates to the advisory committee on immunization practices for the COVID-19 vaccine work group and the larger ACIP on COVID-19 vaccine safety. Through all of these systems and working groups I've discussed, collaboration has been essential. Collaborating with the FDA has been a critical part of the safety assessments of the current COVID-19 vaccine use in all ages. It's extremely important since summer 2020, even prior to any vaccines being under the EUA approved in the US, that we began our planning for implementation of a protocol for the monitoring the safety of COVID-19 vaccines. It was mission critical that the VSD share our working protocols, adverse event outcomes list, outcome definitions, and complex study algorithms with BEST and our FDA colleagues. It was extremely important from the start we synchronized and harmonized our surveillance plans to ensure we were all on the same page and this information was being shared. We established bi-weekly federal partner calls with the FDA and other federal partners where we could discuss preliminary reports, details on our statistical methods, and share chart abstraction forms. I previously mentioned the newly formed VAST work group. VAST served as one of these primary venues for collaborating with BEST and the FDA. It has also served for a platform for us to collaborate with our global vaccine safety colleagues on key issues that impact the benefit risk analysis of COVID-19 vaccinations. There is no question we here at the Immunization Safety Office have experienced the vaccine safety surveillance assessment like one that has never been done before. But all this hard work is not over, and it's going to continue as we begin to work harder as we monitor the safety of booster doses and continue surveillance as vaccines begin to be administered in the 5 to 11-year-olds. All our projects and surveillance efforts will remain in place, and we will surely face new challenges, but I'm confident our systems and our collaborations will help us continue to monitor the safety of COVID-19 vaccines. And most importantly, we will continue our collaborations, our communications with our partners at FDA and BEST, because a comprehensive system for monitoring vaccine safety data informs regulatory decisions, public health recommendations, and supports public health confidence in the safety of COVID-19 vaccines. So thank you.
Great. Eric, thanks very much for those remarks. And next, I'd like to go to Fran Cunningham.
Thank you very much for inviting me to present today. I'm going to give an overview of our vaccine safety surveillance program and very briefly cover VA overall for those of you that are not aware of it, and for those of you that are, you'll have an update, and then spend time on our actual COVID-19 response and our interaction with FDA, BEST, and our federal partners. Next slide, please. Here's an overview of our VA healthcare system. We are the largest integrated healthcare system in the U.S. We have over 150 medical centers and over 1,200 VA outpatient clinics. Our frontline employees are approximately 400,000. Our VA veteran population is made up of about 1/3 of the U.S. Veteran population. So we treat approximately 6.5 million veterans that use the healthcare system consistently. However, we have over 9 million veterans that are enrolled. We are a system that once the veteran comes in, they stay in until death. That does not mean that they do not use other healthcare systems. They do. Our veterans typically have complex healthcare needs, and have multiple comorbid conditions. VA itself is made up of a rich rich data set, and in which we can access through our corporate data warehouse. We also have direct access to our electronic health record, and is the basis of our surveillance program. Next slide, please. So our VA vaccine safety surveillance system is younger than our medication safety system. It was born out of the H1N1 epidemic, in which it was piloted and continued to grow over time. We have two different systems that we use to track adverse events. Our passive surveillance system, where we ensure that voluntary reports are obtained and reported directly to the VAERS system for vaccines, to CDC and FDA, and our active surveillance system that I'm going to spend time talking about today. We identify predefined AESIs that Dr. Lee described earlier, and we do that in collaboration with our federal partners, with BEST as well as with the CDC system. Next slide, please. So here's just an example of what we do with our vaccine surveillance and how it's conducted. We take these, in VA, we use 21 AESIs that we're tracking, of which the definitions were collectively defined with FDA prior to and with CDC. And we conduct near real-time chart reviews when we identify specific events. And these near real-time chart reviews are done to validate outcomes, as well as to do a better job of confirming, of which we do through two methods. We have both a small group as well as subject matter experts that go in and confirm the outcome and the association. And where does it become important? It becomes important at that point in time to disseminate results. And so we have our summary reports, our newsletter, and our RCA presentations. It is the summary results in our newsletter that are internal, but our RCA presentations is what we provide to our outside group, and what we spend time with our FDA colleagues consistently, and with the vast group that Dr. Weintraub discussed earlier. We also will conduct full studies to confirm or to evaluate some of our signals in more detail. Next slide, please. This is just an example of some of our internal reports that go out and what it looks like from a surface standpoint as they're going out. Next slide, please. So I'd like to kind of finish by discussing our collaboration with our federal partners, of which we would not be able to do the work we're doing without them. The work that we are able to conduct from a healthcare system is immeasurable. Having direct interaction, we meet weekly with our FDA colleagues, discussing definitions, discussing methodologies, reporting on things that we may not have thought would have come up, and just discussing that. And also being able to directly communicate with them as things come up from an administrative standpoint with our leadership, having the ability to address that very quickly so that we can respond internally. And we have our bi-weekly meetings with along with our BEST colleagues, but also with our CDC colleagues that are integrated through the VAST program that Dr. Weintraub discussed, that we're able to present our information and share with our outside colleagues. And more importantly, see what they're presenting so we can learn from them as well. One of the biggest things that's really been beneficial for us as a healthcare system, and I don't know if the BEST group realizes how much they have helped us with outcomes definitions and making sure that we can have tools that we're able to validate during our chart reviews. And this happens for both our CDC and our BEST colleagues, so that when we're validating and abstracting that information from the chart, it's valuable to us, but more importantly, it's valuable to them, because then it's apples to apples and oranges to oranges. So as we're presenting the data back, they can use it. So that relationship is priceless from my standpoint. We also utilize systems to help them when they need help to assess signals in our database. And lastly, we enhance evaluations with this collaboration. Has helped us better define some of our outcomes. You're only as good as the database you have, and your database is only as good as the deficiencies you're aware of. So it's really good to have this federal interaction so that when you realize your deficiencies, you can correct them, or you can address them so that the data we have becomes very valuable for us, especially for our veterans. That's who we are here for. That's who we want to make sure we're taken care of. So we want to have the best information we can have to optimize it. And it's working with our federal partners where they can kind of poke holes in our databases and allow us to enhance what we need to do to make our system better, not only for our veterans, but also to help them. So the collaboration with BEST has been so helpful over the years. We meet weekly. It really is like meeting with another team member. We are just, it's, the relationship is phenomenal. The trust is phenomenal. What we learn from them is great. We really do appreciate the interaction with our federal partners and our BEST partners. And the collaboration has only enhanced our response to our veteran population. Next slide, please. And I'd just like to acknowledge our team internally as well as our federal partners. Thank you.
Great, thanks, Fran and Eric and Wheelie for covering a lot of ground on a truly government-wide response involving not only your agencies, FDA and CBER and BEST, and a number of parts of CDC, VA. And you mentioned the tight partnership with CMS and all the outside partners. And we're going to get those outside partners and the non-government partners in our next session. So this one, we had a number of questions that relate in one way or another to what's worked and what was learned. I don't want to say what doesn't work, because this has clearly been an iterative process, as all of you emphasized in your remarks. But what have we learned about ways to make this comprehensive government-wide response system as effective as possible, not just for future public health emergencies where we need rapid evidence from the real world on rare safety effects or other safety consequences, but also, of all of you have emphasized, questions about effectiveness as well. So I want to talk about that, but maybe we could start with just some of the basic data issues and key questions that you're trying to address together as maybe a way to talk through some of these. And I'd like to start with the adverse events of special interest, the AESIs that, as you pointed out, were identified even before the vaccines were authorized based on both suspected concerns, theoretical concerns, et cetera, before the vaccines even came to market. You all talked about a lot of systems that came together to answer these questions, some in the category of, you might call it spontaneous reporting, or I think some of you refer to them as passive surveillance systems, like the adverse event reporting system for vaccines, and then the vaccine VSafe program that CDC set up quickly. It had large amounts of people participating via app on reporting on their own symptoms and experience. But then the active surveillance systems, which, as Fran was just saying, were happening across the VA, across the Vaccine Safety Data Link, across those data partners that Wheelie mentioned, and also Medicare data. One of the basic issues, though, in detecting adverse events and understanding differences between vaccines or not is getting reliable data on who's actually been vaccinated. And I know early on, especially when a lot of the vaccination events were happening in public health settings, big mass vaccination sites, other settings managed by public health and with data systems and the state immunization registries, seemed like there were some concerns about the connectivity of those data to data from people's usual healthcare providers or health plans that might have information through the active systems on what the consequences after the vaccination were for patients. So it's a long way of kind of setting up. One of the data issues seemed to be actual accurate information, timely information on vaccines. You all talked about ways to deal with that, but maybe if you could expand a little bit on how you all as a group thought about that issue early on and took steps to address it together.
I could jump in first from the CDC's perspective. We had a lot of experience with this, with dealing with H1N1. And then I kind of set the tone. I think it was really important, even prior to H1N1, pandemic planning was the top of our minds, even starting all the way back to like 2005, 2006. We knew pandemic planning was important, and I think it's an important thing to discuss as well, of being prepared, pandemic planning, and for us to be prepared to have protocols implemented and ready to go for H1N1, any other future pandemic, and obviously COVID-19. From learning from that, it was clear, state registries were critical, and you had to have linkages with state registries. There was also requirements for, facilities that received COVID-19 vaccine, that they had to participate in certain registries and provide and share data. So VSD, it was very critical up front, and we were fortunate that most of our sites had also established very positive relationships from using flu from previous years with large pharmacies as well, where there were data linkages occurring. So right from the start for us, we were getting data dumps from state registries, bi-directional. Our sites were giving the data to the states were giving data to the VSD sites. We're pulling in data from pharmacies, and we can tell right now the general coverage in VSD is around 70 to 75%. So we've done a very good job at capturing vaccines. Obviously, there's still some that are being missed. You're not going to capture them all. We also allow people they come in for a medical appointment. We allow self-reported vaccinations data that can be entered in, even if it's prior to enrollment. So vaccination is critical component, and it's one of the pieces that also then drives what types of analyses and statistical techniques can you utilize when looking for the safety and assessing the safety of vaccines. Because if your population, if your data source, let's say, doesn't do a good job capturing vaccinations, you need to be very cautious on who your comparisons are and who you're comparing to. Because let's say if you're comparing to an unvaccinated cohort, there's a very good chance that a large portion of those individuals are truly not unvaccinated and might be vaccinated. So it brings up another interesting concept of having multiple comparisons at your fingertips, not just historical backgrounds, not just concurrent, but having different comparison groups available so that if a signal does arise, you can look at different analyses and see how they compare.
And that seems like an advantage of having these multiple analyses going on at the same time, where you could compare. I wonder, Wheelie or Fran, any comments about your experience and the data sets that you were analyzing with this issue of just as Eric said, it's important to avoid the noise, the bias towards zero that can result from mismeasurement of a critical variable like that.
Sure. If I could jump in on behalf of the best. So we recognized that very early on, as Eric and Fran would say, the capture of vaccine. And so one of the slides that I showed actually is very brief on what we're working on, linking the state and actually it's state and local jurisdictions for the immunization registries. We have some linkage already actually, and we'll be using that for vaccine effective studies. We'll be using that also for safety. This effort is of high importance to us, and we also have the support of our center director, Dr. Peter Marks, who've at different levels of outreach and different levels of linkages has, for example, spoken on behalf of these efforts to the Professional Society for Immunization Registries, and that is the AIRA twice actually, to push this forward, and has been very helpful in pushing these efforts forward.
Yeah, sounds like a very important initiative that got a boost with you all working together across public health and FDA and so forth on these issues. Fran, any comments on this from the VA perspective?
Yes, and I think some of our work really originated out of some of the earlier work we did with BEST, kind of identifying where some deficiencies were with our influenza vaccine and with our Zostavax vaccine, in which we had to do a better job of capturing information from pharmacies outside of VA that some of our veterans were having their vaccine administered. And earlier on, there was a direct relationship with one of the pharmacies, and so there was a direct dump of vaccine data from our veterans there. More recently with COVID-19, we get them from multiple pharmacies. So we're able to identify in our database through our immunization and through the record those veterans that get vaccines outside of VA. There is still a weakness if there's a mass vaccination site that is occurring at certain areas outside of the VA. We may not pick those patients up. We are picking the patients up, however, for those that are getting mass vaccinated in VA. There's a system that allows us to identify those patients and make sure we capture. What we do is we go in and we do a random evaluation. We do that multiple times per month, select a random sample of patients to see what's going on, where weaknesses are, to ensure that we're identifying and capturing correctly. So we've done a very good job of doing that during COVID-19 response.
Great to hear about all the efforts on this and the synergies between them. I do want to ask about, several of you all mentioned the so-called passive surveillance systems versus the active ones, the ones where reports come in spontaneously or with, in this case, a lot of encouragement by CDC, FDA, VA, others, at least when there was a suspected adverse event of concern emerging with thrombocytopenic thrombosis, with myocarditis. Could you all comment on how those systems kind of fit together? It seems like there's been a big trend accelerated by all the work that you're doing to get more comprehensive and accurate population-based data. Vaccine safety data link's been around for some time, but it's gotten richer, as Eric described. BEST is relatively new and more comprehensive, and VA as well, seeming to make more use of more longitudinal interoperable electronic data to support these population-based systems. Do the passive systems still play an important role? It still seemed like, and you all talked about this a little bit, that those were helpful in identifying some adverse events of interest early on. But can you talk a little bit about how those systems fit together now and what the future looks like for passive surveillance versus these increasingly complete and larger active surveillance systems?
I'm going to start first.
Okay, go ahead. Yeah, Wheelie.
Oh, sorry, Eric, did you want to go first? Go ahead.
Oh, I could. Yeah, I'll be quick. I apologize. VAERS,
okay.
They're not necessarily my area of expertise, and probably let the FDA colleagues speak more on that behalf. But I think this is kind of a unique situation for some of these passive surveillance systems because of they're doing a lot better job than they probably historically do because of the sensitivity with COVID and the importance of the
visibility visibility.
Yeah, the visibility. I think sometimes you're often worried about the concept of the opposite, over-reporting happening, that there's more reporting going on than under-reporting. But I still think the systems all complement one another very well, because VSD in particular, like our system right now, is really focusing on outcomes that required individuals to go to have a hospital stay or show up at the ER visit. VAERS does a lot better job with local reactions, maybe less severe things, and that's where vSafe comes in as well. vSafe was able to ask, did you miss work? Those are the types of things of primary data collection that that they can do quicker. And also at CDC, we have a huge team set up here of individuals, nurses, and physicians that are contacting individuals, reviewing VAERS reports, discussing VAERS reports, getting as much exhaustive information as they can. So often it's used as kind of the gateway, you could say, and we think there might be an issue with thrombosis, following a particular vaccine. Let's dig deeper in these other systems and start looking at medical confirmation of these outcomes. What is the risk window? VAERS doesn't necessarily do a great job of what is the risk window. That the further you get out from the date of the event, the less likely people are going to be to report an adverse event, let's say. So usually you have clusterings of conditions that occur closer to the timing of vaccinations, where these other systems, VSD, BEST, and the VA, we can look and say, did people have more visits at times closer to the vaccination? Or in the 22 to 42 days later, where there are clusters of visits. So we have a little bit of flexibility, but I think in general, the systems all complement each other well. And the important piece is by using VAST, by using our federal partners calls, we can all discuss these important issues, discuss if risk intervals are correct. Risk intervals is a very important conversation. What do we think is the appropriate interval where we think an event occurring in that time following a vaccination could truly be associated with the vaccine? So I think those are critical conversations and discussions.
Thanks, Eric. And if I could jump in at this point here. So to add on to what Eric said, I personally actually see these two systems, the passive and active surveillance systems, as complementary, and both are needed for the surveillance of this. For the passive surveillance, the VAS, which actually our Office of Biostatistics and Epidemiology at CBER oversees both the passive VAS, working in concert together with CDC, and then the active surveillance is that's actually the best system. So for passive surveillance, and that's with our colleagues at Division of Epidemiology and with Dr. Aaron Nair and his colleagues who oversee that. They've told me, they've mentioned to me that they actually have a lot more reporting this time for COVID-19 due to visibility, but at the same time, it's hard to get rates from that. And so that's when our systems, the active surveillance systems, as you mentioned, population-based systems come in to obtain rates, to compare, to actually make comparisons of whether those are actually elevated rates or are they just what is seen in absence of vaccines, for example. And as Eric pointed out, then actually to go in and provide more information about some of these cases, the risk intervals, certain age groups, or a certain demographic, because some of this information is less, it's not so populated in the passive surveillance system. So I actually do see both, and some of our signals are actually from passive surveillance that we've actually then followed up very quickly.
Yeah.
So I'm just going to wrap up really quickly and talk about the practical aspect, because in VA we have both the passive as well as the active, and we really set up their passive so that we can have a direct conduit to theirs to ensure that whatever is being reported goes directly to CDC and FDA. But what we do is we look at it internally. So it has really helped us assess the reactogenicity aspect of these vaccinations, of these different vaccines, and have us to, it allows our providers to see what's happening in the veteran patient with the different vaccines. And so that passive surveillance really helps us collect information from a reactogenicity standpoint that we really can't get as well with our active surveillance. So that has helped us immensely. And it also helps us to determine what we may be looking at or need to look at in more detail when we're looking at our active surveillance data.
Yeah, it's great to hear those perspectives and how they're, again, kind of aligned and, as Fran just mentioned, interactive across the programs, and with the passive systems, the adverse event reporting systems, and the like, being really good for giving people context about what getting a shot is like, the reactogenicity, as you mentioned, and also it sounds like it's been helpful in COVID as well for identifying potential adverse events of more significance that would be part of that special follow-up efforts in the active systems. So let's turn to those active systems in a bit more detail related to, you've talked a bit about how they've been used for addressing for identifying whether adverse events are greater than background, and steps to make sure that the comparison groups really are vaccinated, unvaccinated, and properly matched, and so forth. But as you all pointed out, these systems have also turned out to be critical for answering very important questions about effectiveness that are just difficult to do in traditional clinical trials. The durability of responses, differences across subgroups, impacts of potential variants and the like. And I'm wondering if you could talk a little bit more about for these uses, or are we there yet? Are we where we want to be as a country? And we're talking about, for example, let's start with rare side effects. We're talking about event rates on the order of 10, 15 per million, with background rates that may be a little bit lower than that, but perhaps in similar magnitude. boy, that suggests you need some really large data sets to be able to do this. And obviously the work that you're doing, pooling different data, if not pooling, or at least parallel analysis of these different components of BEST is helping with that. But are we where you want to be? Or we, clearly we've been able to get a lot of evidence out of the system about rates of rare side effects versus background rates. But I just appreciate any comments on successes and opportunities to build.
Yeah, I think it's a challenge. I mean, the field of vaccine safety traditionally is you're dealing with rare disease epidemiology. You're taught in universities and master's and doctor programs about rare diseases, and vaccine safety usually are very rare. You're not talking 10% rare, you're talking less than 1%. So, and then you're dealing with a completely different beast than drugs, because you're giving vaccines to predominantly traditionally like healthy individuals, let's say, versus drugs that are for certain conditions. So particularly the sensitivity with, vaccines in children, it's a completely different topic, and it makes for a challenging issue because you're dealing with relatively healthy individuals getting vaccines versus the importance of drugs and medications. Growing is important, and there's no question. Like the VSD is always looking to expand, go to different populations, what areas of the country are we missing? Is that particular ethnic group are there racial disparities with adverse events and vaccine uptake? So there's lots of different ways to look at it. And I think a lot of times what we've done, we've had examples where the VSD might detect something and we've exhausted everything we can do in the VSD. So we turn to our colleagues at FDA who can use the Medicare database, for example, and look at larger populations. And it might take longer sometimes to explore, but I think we all have different systems in place that complement one another and well. But then I think it's important that we constantly have conversations and discussions so that when we do reach the limit of what's available and ability of our systems, we have those communications, because resources is always a challenge. There's no question. Yeah. I mean, now in a COVID world, you have resources that are available that traditionally aren't available to you. And I think that's the challenges. And once we get over COVID, hopefully very soon, resources go back to normal, and you don't have the influx of resources that people are ready to commit to vaccine safety, to vaccine delivery, to establishing systems, to establishing, state registries in states that don't have state registries. So it's extremely important to always think and continue to think about the importance. As I mentioned, pandemic planning is one of the most important things, so that we're ready to go. Hopefully for the next one, we were very ready to go to study the safety of vaccines. Hopefully we're ready in the future for disease aspect as well, and we can learn from what happened now from a disease perspective and surveillance. But it's always trying to be prepared, and pandemic preparedness, I think, it's a key component of why it's important for resources to be allocated in that direction.
Yeah, certainly. Other infectious disease trends, rare but important emerging trends like antimicrobial resistance, not to mention all the other threats to population health, where these kinds of data systems are really helpful. Wheelie, Fran, other thoughts on this question of are we there yet in terms of the sensitivity needed and accuracy that can really help make these get to these answers faster?
Brian, is it okay if I go first, or do you want to go? Okay. So I think there are two parts to your question. One is effectiveness, if I recall, and the other one is sensitivity of this. Fortunately, very, very rare at this event. So the first one, effectiveness, yes, for regulatory decision making, we do actually balance both the benefits and risk. Our colleague Dr. Hong Yang in our office and her team is leading that efforts over there with Dr. Forche. And so with that respect, we actually have brought on new partners to help us with the vaccine effectiveness study. You'll hear more of that in the next panel. But in terms of that, it goes back to, again, what is the best infrastructure for that? So what's required for vaccine effectiveness in terms of absolute effectiveness? That would actually, as you alluded to, vaccine captures. In this case, to link then our claims databases to immunization registries, which is what we've done. And those efforts, I summarized that very quickly, but I want to acknowledge that those efforts, it was quite, it is a large effort. It's a parallel effort both from our data partners who've gone out to do that, and also within the FDA, our team have actually gone out in parallel and working together to actually to do that. So that's one part. So yes, we actually have protocol in development. We'll be putting that and executing those. And then the questions for those will be, as we have found with our CDC and VA colleagues, in terms of responding to pandemic, it tends to be rapidly evolving. So in our case, we have to respond to, in this case, would be boosters and pediatrics, for example. And that's the effectiveness part of it. So the safety part of it, of this very, very, very rare event, do we have the sensitivity of that? So we're always looking for larger and expanding our infrastructure, and this is what we currently are doing while responding to the pandemic. One thing though, in terms of what is needed is really the type of, in our case, we went for claims because of the rare adverse events, but we also need claims data that is has a short data lag. So we looked for pre-adjudicated claims, for example, with some of our data partners, and a refresh that allows us to do this type of testing every 2 weeks or every month. So it's certain attributes of claims that we also then seek and look for. So yes, do agree that we continue to expand in terms of response. Francis, pass it over to you.
So in respect to our specific database, and I'm going to address the rare events first, we're only so large. And so we really do depend on our collaboration with our federal partner colleagues and others to be informed about rare events that may not manifest in our patient population. And more importantly, there may be events that may not be so rare that our patient population may not be young enough to assess. And so we look at our partners for that standpoint as well. So if there's a way for us to improve interaction with our colleagues and our federal partners, we can. We can't expand our database any further. What we do have, however, are certain comorbid conditions. We have certain patient populations that we can evaluate very comprehensively from a safety standpoint as well as an effectiveness standpoint. Immunocompromised patients come to mind, especially those with multiple comorbid conditions that we need to look at. We independently are looking at them in a lot more detail, perhaps maybe even more than our federal partners are aware of, that we'll ultimately, that we'll need to make sure that to get that information over to them. That's important not only from a safety standpoint, but from an effectiveness standpoint. And our databases are very well set up to evaluate effectiveness, which has been done in the past. And we don't look at it specifically, but we have colleagues that are looking at it and are able to get information on the vaccines now and are working with our federal partners to ensure that those studies are done. Variants, again, very important, especially in the VA healthcare system with our Million Veteran Program. The ability to look at that becomes paramount. So it's something that we're doing.
Yeah, I would like to spend a couple more minutes on this. We've got a little bit of time left, and thank you all for a fascinating discussion. Wanted to turn to, okay, we talked about rare side effects and the benefits of having sort of larger populations that where consistent analysis can lead to more definitive conclusions, like Fran was just describing from the VA coordination standpoint. For effectiveness, the sample size perhaps aren't so large. They may need to be pretty large if we're talking about specific groups within the population, like pregnant women or people who are living in higher risk settings or have important comorbidities, as some of you mentioned. But if we're talking about causality, hopefully the vaccines have large effects, but if we're worried about things like diminishing benefits over time and so forth, the methods really matter too, has been emphasized yesterday and today. And we've seen a number of studies come out of the programs that you all have participated in. We've seen some from other countries, Israel, England, and it's not so much, I think, that those countries have, everyone in the same database. Israel has multiple health plans and 3 major health systems. England's got a lot of diverse providers all across the country, but they have put a lot of effort into common data models and methods for answering these questions about observational data. What is really a good comparison group? If the people who got vaccinated early were different from the ones, except maybe in some ways that are hard to measure, from those who got vaccinated later, is that going to bias results about effectiveness of vaccines over time? And if a variant does emerge, is that emerging in a representative population? How do you do matching? And as we talked about some yesterday in those contexts, and these seems like areas where sort of more sharing helps, as it sounds like you all are doing across the different programs. And also just want to pick up on a question that chat from Kevin Hayes, who as well has a lot of experience in this field, suggesting about whether it's possible for the common data model code and maybe some of the results or methods leading to the results, if it's possible to get those more into the public domain over time. Again, I think it's all in the spirit of trying to enlarge the capacity and the comparability of results that are emerging from these different but all important and increasingly aligned systems. Any thoughts about this? It's going to be my last big hard question for the session. So in terms of where we go from here.
Could I take a question?
Oh, go ahead. You can go ahead.
Oh, Eric, go ahead, and I'll take the question later on.
Oh, no, I'll promise I'll be brief, because our group really, I think that's one of the unique things at Centers for Disease Control is our group is really tries to focus mostly on the vaccine safety and then hand over, the VE risk benefit analysis to other groups. Now, our groups, our colleagues at all of our sites are all heavily involved in VE vaccine effectiveness work. So the VSD is a rich data source to still use, and we do have some mechanisms in place to do VE work. So I think what you're highlighting as well is the unique challenges with assessing, not just for VE work, but also safety, special populations. And I think that's where it kind of answers your other question as well about opportunities for growth. And I think that's the most important piece for where systems can grow or could grow, is when it gets to special populations. Often you're talking about myocarditis is a perfect example where you're thinking, you're not looking really at all ages. You're focusing on, let's say, 19 to 39-year-olds, 19 to 25-year-olds, where once you start looking at systems and you start looking at smaller age groups, the numbers get smaller and smaller. So that's where it's really important to think about growth and then be able to also complement. I think risk benefit is not even just about VE. It ultimately comes down to what is the risk benefit. And I think that's where it's extremely important to be tied in with a group who can help. We can feed them, this is what we think the risk is, and then they can turn around and say the risk benefit analyses as well, based upon having risk data, vaccine effectiveness data, and then taking that all into consideration, truly assess the risk benefit. And like we mentioned, the vast majority of things we're looking at are extremely rare. So often the benefit outweighs the risk predominantly for most of the population.
Thanks, Eric. Great response. I'm not sure, Wheelie, were you next or?
Yeah, Fran, is it okay? Sorry, I'm going to go ahead. And there are 2 parts to this. I'm going to first touch on what Eric just mentioned about risk benefit. So in terms of communication of these results, there is a risk benefit part of it, but we are also looking to analysis and discussions about analysis. For example, myocarditis, that's the risk. Post-vaccination, and you compare that and do similar analysis, what's the myocarditis risk after COVID-19 or SARS-CoV-2 infection, and compare those 2 together, and then communicate that out. So yes, there is a risk for vaccines. This is the absolute risk, and then compare the absolute risk with, had you actually been infected with SARS-CoV-2, and what's your myocarditis risk there, which actually is higher. So these are the different type of analysis that we are also thinking of doing to communicate the risk of these vaccines. That's one. The second thing is what Kevin was asking. I didn't mention, I guess, in the last slide that I had, that we have QR code and our best site there, where we publicly post our protocols, publicly post various of our materials there to the public once it's released. For example, for the ISS linkage immunization registry, information registry that we have, we don't just link it. We also we are now reviewing and clearing a final report that explains how we did it and what are the steps in terms of quantifying what else do we think is missing after linkage from external source in this particular case. Trying to pass it to you.
Great, thank you.
And I'll wrap up very quickly. And because again, we are a system that has both the ability to look at risk and benefit. We are, and we do, and we're setting up to do more evaluations accordingly. I think one of the things VA has, and it's not my story to tell, but we have a very good Office of Research and Development that has spent a lot of time just working on independent methodologies. And those independent methodologies are now being applied across the board, not only to medication, but to vaccines as well, so that we can enhance methods from a vaccine effectiveness standpoint and look at things accordingly. So more to come.
More to come. Well, I want to thank you all. We're about at time now. I'm going to give you one last quick comment, but I did want to observe just how far these kinds of surveillance and tracking and evidence development systems on both safety and effectiveness have come during the course of the pandemic. I'm going to go back to what Eric said earlier. So lots of momentum here that is, unfortunately being driven by just the tremendous impact that COVID has had nationally and globally. My hope, as you all have said, is that this effort will continue. That it's not going to get a new motivation from some very worrisome new variant emerging or something like that, but that we are going to be able to turn this into an ongoing infrastructure that could really answer, if you think about it, a lot of large scale questions, not only about the future effectiveness and safety of vaccines against emerging threats or even against the flu, but also potential implications for other biologics and other treatments that are a big part of Sentinel. So thank you all for all the work. Any final comments about, as you think about the continuing to build on the progress here, that outside world stakeholders should know or could do to help advance this work?
Could I?
No, I think you covered everything, and I'll just add, I think the last piece I'll say is accuracy of exposure data is one of the most important pieces. I mean, our group is tasked with vaccine safety, so the quality of the vaccine data is of critical components. And when people, even outside of this realm, when hopefully we get over COVID and they're documenting HPV vaccines, they're documenting Manuscript vaccines, they're documenting influenza vaccines, how important it is to get that information correct and enter it in their systems so they can be properly assessed and studied. And obviously it's of the utmost importance when you talk about medical devices and drugs as well.
Yeah, and connect that between public health and healthcare systems. Thanks, Eric.
I will close with this, that it actually has been my honor to work with our federal partners. We actually have worked very closely together, responding to the ever-evolving, changing, being open. There's a lot of trust. And it's just, I feel we actually build infrastructure for future relationships in terms of vaccine safety.
And I would like to close emphasizing what both Eric and Yuli said. One, the accuracy and encouraging. We've really been able to encourage our providers based on this COVID-19 response, how important it is for entering data. Having a system to validate, even the entry of data that's going in there from a vaccine standpoint, as well as outcomes. I think that becomes very important and shouldn't be overlooked in various healthcare systems. And then to state what Yuli said, our relationship with them has been priceless, and our veterans are better for it. And we're better as independent researchers in surveillance and pharmacovigilance effort. All of us are just better for the current relationship that we've had, and it's been a pleasure to work with them.
Well, I want to thank you all for this session, especially taking time out. I know your days have been very, very busy, even more so than usual over the past year. So thanks for taking the time to spend so much effort explaining where you are, where you're headed. And I hope we can, as you all said, continue to build on the progress here. And shown a lot about what can happen, so a lot more that we could do to build out these systems further, but really appreciate the efforts. And for all of you who are joining us today, we are going to take a break now until 12:40 Eastern time. We went a few minutes over with this important session. When we get back, we're going to talk more about some of the issues related to BEST and improving these both surveillance and effectiveness systems from the perspective of the partners outside of the federal government working on the BEST program. So I hope to see you back in about 26 minutes. Thank you. All right, good afternoon or good morning, depending on where you are, and welcome back to our second day of our 13th annual Sentinel Initiative meeting. For this session, I'm very pleased to close out this meeting with a discussion from the collaborators of the BEST, from the partners in the BEST collaboration. We're going to build on some of the topics we heard about in the last session related to the role of Sentinel and BEST and related activities in the response to the pandemic, again, keeping with this focus theme on understanding vaccine safety and effectiveness. As you heard in the last session, a number of challenges there, getting exposure data about vaccination right, being able to track increasingly large populations of patients over time and reliably in terms of data, understanding the data, and applying effective methods for evaluating both comparisons to background rates of adverse events of special concern, as well as developing needed evidence that is hard to capture in clinical trials around subgroup effects of vaccines in populations of key interest, around issues like vaccine durability of response and impact of boosters, impact of variants, and the like. So now we're going to hear from a broad group of the collaborators in this effort outside of government, along with a perspective from the Biologics Center as well. The panelists for this session include Rich Forshee. He's going to kick off with that CBER perspective. Rich is Associate Director for Research for CBER's Office of Biostatistics and Epidemiology. You heard from him yesterday as well. Next is George Ripsack, who's the Vivian Beaumont Allen Professor and Chair of the Department of Biomedical Informatics at Columbia University. Then John Seeger, Chief Scientific Officer for Epidemiology at Optum. Tom McCurdy, old colleague of mine, Senior Research Director at Acumen LLC and Director of the BEST Data Coordination Center, and Bradley Layton, who's a Senior Research Epidemiologist at Hca Health Solutions. So a lot of partners in this effort to expand out the capabilities and the comprehensiveness of this very important effort around real-world evidence on safety and effectiveness in the pandemic. And again, just like last session, I hope we'll have a chance to think about the implications that this work makes as a foundation for more effective real-world evidence generation on Sentinel-related issues of safety and effectiveness of medical products for the future. So I'd like to start with turning to Rich.
Hello, everyone, and thanks so much for allowing us to be here today and to talk about the work that we're doing with our BEST and our partners in BEST. I'm just going to say a few words to kick things off. I do want to set a little bit of the context here. We really started working back in January of 2020, before there were any reported cases of COVID-19 in the United States, to try and make our systems as ready as we possibly could for the possibility of a major pandemic in the US. We were working to make sure that the data systems themselves were ready. We were working with other stakeholders to get all of the coding in place so we'd actually be able to identify first COVID-19 diagnoses, and later on to have codes in place for the various vaccines that were in development. So I mentioned that just to say that this has been, we've been at this for nearly 2 years now in terms of trying to use our systems to respond to the public health emergency that we're in. During this time, we've been working on making our systems faster and better. We wanted to work with our partners to reduce the time lag from seeing the data and being able to use those in analyses. We worked very hard to set up a rapid cycle analysis to provide early screening for a long list of potential adverse events of special interest. Later on, we've turned our attention to inferential studies, and as we'll hear in some of the discussion, developing some vaccine effectiveness protocols. So it's been a busy couple of years, and I really want to thank all of our collaborators in BEST. Frankly, we've pushed them pretty hard at some times in order to try and get data that was really critical for public health. And I just want to say they've been great partners in working with us in order to meet the needs of the public during this unprecedented crisis that we've been dealing with. My partners have a lot to say in their presentations, so I think I'm going to just end my opening remarks there with, again, expressing my gratitude to our partners for working with us. And we can go ahead and move on. I believe George is going to be the next speaker, and let him start his presentation. Thank you very much.
Thanks very much, Rich, and thank you for inviting me and being able to present today. I'm going to be presenting the FDA convener, BEST convener component, which hosts and convenes meetings, does methods development, conducts training, and works on dissemination. Next slide. I'm from Columbia, representing the Observational Health Data Sciences and Informatics, or Odyssey initiative, which is the convener contractor. The physical contractors are Columbia University, Northeastern, UCLA, and Hopkins. Odyssey's mission is to improve health by empowering a community to collaboratively generate evidence. Has over 2,000 collaborators from 74 countries. Its federated database, which is not the best database, it's a separate database, is over 800 million unique patients at this point in time. Next slide. As you can imagine, with COVID, there's less meetings to convene and training sessions you can do, but we had a wonderful virtual seminar series on vaccine safety with world leaders in researchers in vaccine safety, with attendance over 200 and people watching from all over the world. Next. I'm going to talk about our research, methods research right now. There's two kinds of labels. The BEST means that it was basically commissioned by the FDA for the BEST initiative. As a convener, we're also here to collaborate and coordinate. So I'm also going to show some Odyssey methods in clinical research that were informed by the public BEST progress and reported back to BEST. Next. One thing we did was comment on the FDA methods protocols. There's two examples, and we appreciate the opportunity to do that. Next. So one big study we did for BEST was sensitivity of background rates, historical rates, to study design. As the first thing you do as you see reports coming in is compare it to historic rates. And the question is to quantify what is the bias for differences in study design with a pre-specified protocol. So there are things we know about age, sex, race, chronic disease, health-seeking behavior like vaccines, but we quantified it, and also things like time at risk, what year, what duration, and something I'll call anchoring, which you'll recognize but isn't well written about, which is you have a group that was vaccinated, then you have the control group, and if they weren't vaccinated, what's the index date for the beginning of the time at risk for that group? Where they had a outpatient visit, was it a healthy visit, or was it a random date in their progress or another vaccination? Next slide. We had a lot of results. I'll just show two. The first graph shows myocardial infarction by age. We all know that age is going to affect that. My point here is that it varies by 1,000 in different age groups. What that means is, yes, you need to not only correct for it, but you got to correct for it correctly. And if you get it wrong, you can easily get a rate ratio of 1.5, given that the difference between the groups is a factor of 1,000. But the interesting finding that I think is largely not talked about is the anchoring on random date visit or healthy visit. For longer times at risk of a month up to a year, we can still get effects up to almost a rate ratio of 3, which is important. And if you look at something like anaphylaxis with a time at risk of 2 days, you can get near 100-fold risk depending on which anchoring you use. Next slide. Dr. Cohey earlier today referred to some work that they did with Odyssey on coagulation. These are the data that we supplied to the EMA on March 18th in their determination about AstraZeneca. We basically what we took is what we learned from the previous thing, generated the protocol and generated the evidence. This is an Odyssey study. What's fun about it is that the work that we did, commissioned by BEST, produced methods that were directly resulted in the protocol that was used to influence the vaccination of millions of people. Next. Another Odyssey study, we often do method studies. So this is comparing for vaccine safety, case control cohort, historical comparator, and self-control designs on, say, 70 negative controls. Now, it turned out that self-control designs did best in this test, but more important than that is the huge type 1 error that we found, which outweighed all other factors. In other words, we worry about things like sequential testing and alpha spend. But if you don't get this right, then it's far bigger. Odyssey, it solves it through calibration to reduce the type 1 error, but there's no free lunch on that. Next slide. The last Odyssey study I'll show is vaccine effectiveness. Look at it as a proof of concept done to complement other studies. We're using at first the Columbia data because it's linked to city and statewide vaccination records, so we know who didn't get a vaccination, but then it's expanding to other Odyssey network members. Our goal was to look for biases and week-by-week analysis. A few findings. First of all, anchoring that I mentioned matters. In fact, the anchoring for influenza vaccine is quite different. In fact, the opposite than the anchoring for COVID. COVID was best linked to a random date. COVID diagnoses can have a high false positive rate. Like 70% of times in the first week after vaccination due to rule out and due to previous COVID. And there are other week 1 biases, the first week after vaccination, where people who have the vaccine assume their symptoms are due to the vaccine, not COVID, found that on chart review. And so that's the graphs on the top, you see week by week, week 1 through week 6 for diagnosis and hospitalization. The first week is wrong, you can see there is biased. But the 2nd, 3rd, 4th, 5th, and 6th weeks quite well match our expectations of the effectiveness of vaccines. This is combining both the Pfizer Moderna vaccine. And then last, if we look at fully vaccinated and compared it to randomized trials, you see that our results, given the corrections that we learned from our bias study, exactly match the randomized trials. I mean, for the Janssen vaccine shown at bottom, our sample size was too small, but otherwise we're getting very good results. Last slide. And then as part of our next best study is Bayesian approach to signal detection. That is, we're measuring a lot of things at once. We have sequential testing, we're looking at different databases, potentially different methods, and different outcomes. How do you combine all that and not ruin your type 1 error rate? Also looking at rare outcomes and subgroup analysis, again, using Bayesian analysis. And I will stop there.
Great. George, thanks very much. Covered a lot of ground there and a lot of good references too. So thank you. Next is John Seeger.
All right, thank you. I'd like to say hi, I'm John Seeger, the Chief Scientific Officer for Epidemiology at Optum, and I'd like to share with you some work my colleagues and I are conducting in conjunction with FDA as part of the BEST initiative. And this photograph on my slide is taken out the window of the office for Optum in Boston. And it was at a time when Fenway Park was used as a mass vaccination site. And I use this as a reminder of the purpose of my presentation, but also as a foreshadowing of some portion of my presentation to come. Next slide, please. Okay, just a bit about Optum. We are a subsidiary of United Health Group, one of the largest companies in the United States. As such, we are business associates with a large health insurer. The Optum claims data comes from this association. We also have access to a large electronic medical record network. Accordingly, we sit within an environment that hosts the data that we actually use for the research that we conduct. And this insider access to the data allows us to do things that would not be possible from the outside and also expands the utility of the data. So given the time constraints of the presentation, I'm going to just present two stories that help exemplify how we are making use of the data in a way that enhances its value for public health surveillance. The first story is about timeliness of evidence. We've heard this a few times already today. And the second is about something that comes up on the penultimate slide, and you'll need to stick around to hear about it. So without a doubt, this audience has a good understanding of the claims databases. And as we know, data has a substantial lag from the time a patient sees the provider until the service claim becomes available for research. And this nice slide from Sebastian Schneeweiss shows many of the steps involved in taking data from the patient to a research database. And what we have done within Optum is set up a data feed that bypasses the last two steps of the process shown in the figure, which means that the data comes to us and can be incorporated into research with a much shorter lag than is typical. And we have just conducted surveillance on COVID-19 vaccines that included data arising from patient visits at the end of October. We are on November 9th today. And so this shows you how short the lag is that we are using in some of our research. It's getting close to sort of the theoretical minimum of the lag because of the time that a provider takes to submit the claim. Next slide. So the rapid cycle analysis is a concept that has also been identified a number of times today. And it identifies people who receive the COVID-19 vaccines and then identifies the outcomes of adverse events of special interest that occur following that date, and then compares those to some background rates. Next slide, please. And this is not our data. This is from a European consortium. Our data is going through the publication process, but it's similar to this. It shows you what background rates look like. It's highly stratified by age or sex and calendar time so that you can make appropriate comparisons. Next slide, please. This is some results from a recent presentation that Hui Li gave to the Vaccine and Related Biologic Product Advisory Committee, showing the size of the database that Optum brings, and it's sort of about 5% of these pediatric populations, and then a number of this case Pfizer doses given. And you can see that nobody in the younger age categories received it. Next slide, please. So then now we have a signals where we do the max SPRT assessment of the observed outcomes relative to the background rates. And you can see, there were no signals for the Pfizer vaccine except for anaphylaxis, and that's something that was expected. Next slide, please. So as has been mentioned in my foreshadowing at the beginning, moving beyond the surveillance, the rapid cycle analyses, you start thinking about customized epidemiologic studies. And often these will involve a comparison of concurrent cohorts. And these concurrent cohorts are vaccinated and unvaccinated. It has been mentioned, how do you know that the unvaccinated group is actually unvaccinated? Because a lot of people might have gotten their vaccine, such as at the mass vaccination site, where it didn't get submitted as a claim, and you can misclassify exposure in ways that can be quite detrimental to the inferences that you'd like to draw from the study. And so we are going through a process of bringing in state registry, working with our colleagues at United Health to do this. And we've already brought in enough to do a reasonable analysis, but more are coming in every week. Next slide, please. And with that, I'd like to thank you all for your attention, and also I'd like to thank my team at Optum, without whom this would not be possible. And I'll look forward to following up on any questions in the Q&A portion. Thank you.
Great. Thanks very much, John. And next is Tom. Good to have you with us. Tom, I think you may still be on mute. All right, just checking on Tom's connectivity. Tom, you there? I see you.
There we go.
There we go. All right,
sorry,
thanks.
I had a slightly different order than you had, so no.
All right, well, thanks for rolling with it.
No, actually, no, you were right. I was off. Sorry. Okay. Purpose of my talk today is that my aim is to basically provide an overview of the use of the best data network as it really was applied during the COVID-19 pandemic. I should note that Rich mentioned that the network had been started to develop a couple of years ago. That's true, but in fact, there was a ton of work that was done well before that with flu vaccinations to essentially work on near real-time, rapid cycle sort of surveillance of the flu vaccine, and all that apparatus was moved over to COVID kind of immediately when it kind of came up. Next slide. So just to give you a presentation outline so you can follow where I'm going, I first want to give a quick overview of the best data network. And then I'll talk about the real-time monitoring of COVID-related measures during the pandemic that actually took place. And then I'll talk about the near real-time surveillance of the COVID-19 vaccines that actually really took place. And then I can talk a little later on about where it's going. Next slide. This is an overview of the best data network as it currently exists. It shows the various partners. There are the 3 government partners of CMS, the FDA, of course, and CMS and the Veterans Association and CDC. And you can see there are a variety of academic partners that are doing a variety of research on this. And then the partners, the best data partners that I'll talk about a little more, that play various roles in the best sort of system network. Let me note there are kind of 3 principal areas of work and studies that the best network does. The first is kind of a near real-time monitoring health events and outcomes. It's for both monitoring the incidence of disease and safety and effectiveness of the various kind of treatments. The second one is a rapid response initial exploratory investigation. So this is kind of what you would call signal detection, where a variety of measures are monitored to detect whether elevated adverse events occurred. And that also partly includes rapid cycle sequential testing of signals to the extent that that's feasible at that time. And then the third one is the support of CBER and FDA in producing FDA position reports and peer-reviewed scientific epidemiological studies. So the notion is there's first monitoring, there's surveillance over an investigation of potential signals. And then the third is deeper studies after that. Once again, considerable capacities existed and were implemented at the onset of the COVID pandemic. So what I'm going to talk about was what was actually done, not what was planned. There are 2 really dimensions of the best system I want to talk about. One is the distributed data network, and the other part is the near real-time monitoring of health events developed through the surveillance of the safety of flu vaccines, but is now being implemented and now also being implemented in the best data network. Next slide. So this is the best data network really has kind of 2 aspects with regard to the surveillance. One is kind of a distributed data network. And the other one is a real-time monitoring sort of system that's using the Medicare data. The description I have right here describes the best distributed data network. And I should note that Acumen kind of is the best data coordinating center. So we're the group that coordinates these partners in various ways. And they each use their own sort of data sets. The Blues Health Intelligence one that I mentioned there is really actually a data set that's used by Acumen as one of the partners. So we both serve as a coordinator and one of the data partners. There's CVS Health, there's HealthCore IQVIA that uses the Anthem data, IBM Market Scan, and then Optum that uses United data, which John just sort of mentioned. And this distributed data network works very much the way you're familiar with the way Sentinel does. There's protocol development, but you get the background rates. There's people are coordinated on the same sort of thing. Data is organized accordingly. There's distributed when the center distribute standardized coding packages, and just conduct a real analysis, etc. And also, there's a medical record review that's developed on this, where to the extent there's relatively rapid within a few weeks of being able to couple months to be able to acquire medical records, at least in the Medicare data, and all this is being expedited on all the rest of the data sources as well. So results in this sort of system are kind of available. The claims delay varies a fair amount, as you mentioned, as John mentioned. United was originally about 1 to 2 months. They're now much more rapid, so that's good. The Blues Health Intelligence is slower, but basically everybody's working on trying to speed it up. But for the most part, results in this system can really are available within weeks or a short number of months, depending on the issue. Next slide. So Acumen serves two roles. We have two hats in this. One is, as I mentioned, we're the best data coordinating center on behalf of CBER. And the coordinating center, as I just mentioned, is really for commercial claims databases. It coordinates with best data partners. Once again, in a familiar that everyone understands and has seen with Sentinel. There's a common data model, in this case OMOP, and coordinates research activities, or basically is really the group that CBER works through to make sure everything's coordinated and they get the kind of information they need. That's the first role of Acumen. The second role of Acumen is Acumen maintains large bodies of databases and analytical capacities directly supporting CBER in its post-market surveillance activities. Next slide. Acumen has been working with CBER for 15 years now to develop this sort of capacity. We have Acumen maintains unified data center. Let me just note that Acumen has 100% of the Medicare data for fee-for-service, which updates daily and weekly for the Medicare Advantage and drug claims. It's got 100% of the Medicaid data that's updated monthly, and it has the assessment data. And what's important here, it has a listing and fair amount of clinical information on all the patients in nursing homes and home health agencies. So let me just note that the data that was used here for the surveillance is called the shared systems data. It's the data that directly comes from the MACs, the claims processing organizations. So we get those data updated daily, and information for those claims is useful after about 10 days after service. It depends on whether it's an IP claim, inpatient claim, or professional claim, but the information that comes in is pretty rapid. So after about 10 days from the service, we have a sense of about a 40% completion, depending on the type of claim. Can be even a little more than that. And at that point, we are able to start given the modeling that we're doing, we're able to start doing forecasts within a week or a couple of weeks. And really the main reason the 10 days comes in is the delay from the end of the week, from beginning of the week. So it varies by day. And there's been a large variety of analytical tools that were developed for Medicare data to conduct real-time monitoring. And as I mentioned, most of this was done for the flu vaccine. The flu vaccine safety had been done for the decade before, and CBER essentially leveraged that capacity for this. Next slide. Now let me talk about the real-time monitoring of the COVID measures that was done during the pandemic. So I'm not talking about what will be done, but what actually was done. I mentioned this already, that CBER already supported development. The CBER, this capacity was immediately implemented at the onset of the COVID vaccination, or the COVID pandemic. And essentially results were generated in March. So a lot of the stuff I'm going to talk about was when the pandemic was coming along and CBER wanted to institute the center's activities, that was basically what was done. And I'll give you some description of the sort of results fairly quickly in terms of what was developed. CBER probably monitored something on the order of hundreds of measures, and to the extent they wanted to look at a different measure, we could do it fairly rapidly. Next slide. This gives an example of the sort of capacity that CBER had at that time. This basically shows there's two curves here. One is the process claims, and the other one is the probable claims. By the probable claims, those are the projections that are used given accounting for the claims delay they're inherent in the data. So essentially the rates are adjusted for depending on what the claims delay history are for those various types of claims. And by pulling down these menus, one can kind of look at, I'll give you an idea of what you can look at, but it could be broke down by, certainly by region, by demographic characteristics, by health characteristics, because with the Medicare data, we can know who has cancer and know who doesn't, and knows who has respiratory conditions based on their health history. So these can be broke down in any way. It's broke down by the state. Next slide. This was the first one I gave you was for the nation. This was done for the states. I think the state happens to be Wyoming, but it's the same sort of picture. Yeah, you should note these forecasts. There were new patients in the last 7 days. So essentially there's an average over the last week, and that's how fast the information was kind of accumulated, albeit somewhat noisy. Next slide. This shows the national maps that were available for CBER, and in fact, a lot of the capacity was developed here was actually used by HHS leadership. Next slide. This capacity allowed them to break things down by states and counties, so they could look at, in this case, there were probable COVID cases. And I should mention the way we measured probable COVID cases was any medical claim that indicated a diagnosis that suggested COVID. So that's the way it was measured in this case. Next slide. The COVID illness can be broken down into a county, but we also had capacities to go down to the census tract. Further, we had capacities to actually look at what was happening at individual hospitals, and that was done in real time. So CBER did have a fair amount of capacity to be able to track the incidence of the disease. Next slide. This quickly shows the maps that you know. I think this was the county of the census tract that essentially shows exactly what the evolution of the growth of respiratory diagnosis was that CDER was looking at. Next slide shows tracking was also hospitalizations that they were interested in. So this was done at the hospital level to show how many individuals were in the hospital with various respiratory diagnoses. Next slide. This shows how, in fact, the treatments were tracked. This one is for this year, but it was broken down by demographics, but whatever other sort of measures they wanted. Next slide. Now let me talk about the near real-time surveillance of the COVID vaccines, because obviously that's one of the main areas of interest. So CBER was ready to start tracking the safety of the vaccines the minute they came out. In fact, some of these results were really used during the trials to help NIH select who was in the trials. Next slide. This shows the CBER's activities of the monitoring of the safety of the COVID-19 vaccines. Essentially, they really, as I mentioned before, they really did this in 2 phases. They had background rates for various adverse events. There was statistical monitoring on a daily basis, and a lot of the results I showed you before were done daily, where updates were done every day. And then there's capacity built in here for doing various kind of max SBRT tests to test the importance of elevated rates. And this could be done, as I noted, this could be done broken down by geographic, but also by any sort of health set of circumstances. The Medicare data is pretty large, and to the extent that FDA wanted to look at particular segments of the population based on health characteristics, those could be readily done in a sort of capacity. That's what was developed. Next slide. This is an overview of the inferential kind of analysis impacts. There was a fairly rapid turnaround of the safety inference analyses. Comparative analysis were done for various health conditions. To the extent that observations showed up in VARs and needed to be in some sense validated, this capacity was done there. And this was these results, and along with the distributed data network, sort of results were used for the vaccines and related biologics product advisory committee in the assorted decisions they were making. So this data was used in real time for the sort of decisions that CDER needed to make along the way, as you guys well know. There was also a variety of epidemiological and effective inferential analysis that have been done by FDA to the extent they need to turn their direction in a fairly quick manner. This sort of thing can be done in this capacity. There's a lot for us to develop, but this is kind of the direction that's really to develop the same sort of capacity with the Medicare data that in the commercial side that we're doing. And next slide. That's all I have.
Great. Thanks, Tom. We're going to come back to discussion in just a few minutes. And next is Bradley.
Yes, hello. Hi, good morning, good afternoon, wherever you're joining us from. My name is Bradley Layton from Hca Health Solutions. We are relative newcomers to the BEST family, but we are very happy to be here. We were invited to collaborate with CBER and with other BEST partners just a few months ago, specifically as attention was turning to the more inferential studies of COVID-19 vaccine effectiveness. And so we are in the process right now. We won't necessarily be able to discuss too many details as the protocol is still being kind of developed and formulated right now, but I will talk to some larger themes and some of our process and how that has worked within the BEST system. Next slide, please. So as I mentioned, we were brought on board a couple months ago to look at various different aspects of evaluating the real world effectiveness of COVID-19 vaccines within the BEST data system. We are scientific partners who were not bringing a data source, but we were asked to lead the development of this protocol and to collaborate with our other data partners and research partners in regular scientific working group meetings to bring this the protocol forward as quickly as possible. Next slide, please. I think that's where, yes. So this has been an interesting time to do this, as obviously in the last couple of months, there have been a lot of key changes in the COVID-19 vaccine landscape. So when we first started talking just not long ago at all about this project, kind of the key scientific question was to evaluate potential waning effectiveness over time, with the intent that might inform a future decision about the potential need for booster doses. Obviously, that ship has sailed as those authorizations and that science moved very, very quickly. And so just the landscape of exactly what aspects of vaccine effectiveness are of greatest interest have changed very rapidly. On the right, I have a plot there of the COVID cases and vaccination over time, marked with some kind of key milestones that indicate where things potentially could be different. As I mentioned, there was an interest in evaluating waning effectiveness over time since vaccination. There's also with different waves and variants, interest in evaluating effectiveness against different variants at different calendar times. And we've introduced new priority groups and booster doses and mixing and matching and pediatrics. And there's just the situation keeps snowballing and moving forward. And so we have been really engaged with this very collaborative and fruitful group as we're trying to determine the best ways to approach all these questions that are changing quite rapidly. Next slide, please. So as I mentioned, maybe the first and key task we had is actually determining what are the key research questions. And obviously, CBER came with regulatory priorities, but then we discussed also, what are the actual relevant questions facing individuals who are trying to make decisions and their providers, and then what data do we need to inform those decisions, and then determine really, can we answer those questions in the real world setting using the data that we have within BEST. And that was a very interesting conversation. And those have evolved over time. As I mentioned, we're developing a protocol. We're a few drafts into it at this point, and at times things have had to be set aside or new things be introduced or start from scratch in some cases as the situation has changed. Next, please. So as scientific collaborators, our primary goal is to really champion the use of valid methods in approaching these issues. And as several of these issues have been discussed already, but obviously there's very strong confounding in many cases between vaccinated and unvaccinated individuals, and that has to be accounted for both in study design and analytic method. And so evaluating what study designs can work in these settings has been key. As I mentioned, there are multiple timescales involved in some of these studies. As we're interested in waning effectiveness over time, effectiveness in kind of a Delta era versus non-Delta era, as we're looking at booster doses. All of these introduce multiple different timescales, and so selection bias, immortal person time bias, and other biases can very sneakily introduce themselves into a study design. And so as we have been working with our collaborators and CBER, we had to ensure that valid methods are brought to these inferential studies. And also one thing that we have championed is as you noted in the slide before, obviously we have had multiple waves of COVID since vaccines were introduced. And so the overall incidence of COVID has changed quite a bit throughout the study period. And the vaccine effectiveness, the traditional vaccine effectiveness measure, is a relative measure of effect. And so if we were evaluating how that relative measure could change over time, we felt it was crucially important that we also introduce absolute measures of effect also in order to contextualize any potential changes in vaccine effectiveness that we observed. And this has been discussed, widely. There are also well-known data issues. I didn't want to steal John's thunder, but he introduced it. But there have been some exciting developments also in linkage of large claims databases to immunization registries to bring all the necessary data in place to evaluate COVID-19 outcomes, validly understanding who is vaccinated and who isn't. And so we have been really excited to work with our collaborators within CBER, within the BEST initiative on these issues. Next slide, please. But obviously, I think maybe the theme of COVID over the past couple of years has been that flexibility has been required. Obviously, developing a large, public protocol is a long, involved process. And as we have gone through this, with multiple collaborators and multiple rounds of revision, like the nature of COVID vaccination has changed as new vaccination strategies have been introduced, new doses, new populations. So as we're moving through this document, we realized that we had to allow for flexibility in this document. And so we tried to develop it in a way that it was somewhat modular, that there was a phase where we primarily looked at descriptive characteristics and seeing what sort of strategies are we actually seeing in the data, and then using that data to inform the inferential stages. And so this has been really an exercise in flexibility as we have the range of questions has continued to expand over time. And then, as I mentioned at the bottom, the data availability has also increased, which has really opened up opportunities to do research like this, which may not have been possible just a couple of months ago, as we're all struggling with how to accurately define unvaccinated individuals, which is really key in these contemporary comparisons of vaccinated to unvaccinated. Next slide, please. And that is my end here. I look forward to our panel discussion here. It's been a great experience working with CBER and the BEST collaborators. And yeah, we look forward to our conversation.
Great. Thank you all very much for the presentations, covering a lot of ground there in the many activities that have evolved rapidly as part of the BEST COVID vaccination surveillance efforts. And I hope we can look both more closely at this and look forward from here to with the goal of continuing this momentum. Just to note, we're running a little bit behind time. We're scheduled in the session at 1:40 Eastern time. If those of you who are here can stay with us a few minutes more, we did build in a little bit of a cushion if we need it. I think you all saw the discussion questions beforehand, and they're all very much in this spirit of what have we learned about what's working now in terms of an effective surveillance system, effective evidence development system related to key issues like rare side effects, effectiveness of vaccines over time and across different groups. But also where do we need to go from here? And maybe we could start with that general topic. I'll try to take you through a few examples to play this out. We've heard about a tremendous amount of work that's gone into getting a range of data sources together, the building on the work that Acumen had already done with Medicare data, for example, that Tom reviewed, bringing in data from Blue Health Intelligence, Optum, other sources, Market Scan. So that would seem to be pretty good coverage of both the Medicare population and the under 65 population. Are there gaps that you see, or do you think we're almost there in terms of the population that's covered with this capacity for looking at claims, but also increasingly looking at integrated electronic medical records and other supplemental information? I'm not sure who wants to take that. Maybe one of you who feels like we've made a lot of progress, but there is still a little ways to go.
So this is Rich. I can start addressing that question. So first of all, I think we have put together a system that covers most of the subpopulations in the United States that we're going to be interested in. Obviously, even with a large network of data partners that we've put together, when you start getting to some small subpopulations, such as narrow age ranges, or when you have to stratify by multiple characteristics, you do still get into some small number problems. But we really are covering a very large number of the people in the United States across the full age ranges. So I think we've made a lot of progress there, and that's where I'll stop.
Yeah, it really seems like it, and appreciate any comments, especially for those of you who are working with commercial data sources. Do you have access to the clinical data you need? Is that an area for further progress? And I'd like to turn to Medicaid as well.
All right, so this is John. I'm happy to say a few words, about what we have in the Optum data, and certainly Optum is really large in terms of the data resource. So we have representation from essentially every state in the country, and then we have the ability through our business associate relationship with the parent company to access medical records, and that helps us fill in some of the missing pieces of information of, is this claim for myocarditis, pericarditis, an actual clinical case of myocarditis or pericarditis? So I think we have, the ability to address a lot of the limitations, and but as a commercial insurer, we are mostly employed people and their dependents, and so there's some portions of the population that aren't present within the Optum data. And so that's why it's important to be a part of the consortium of research sites.
Actually, Mark, I would note that I think one of the biggest holes we have really in the data is the Medicaid data. And the part of the challenges with the Medicaid data is the data that comes through CMS is too slow. But the other issue really is that Medicare now is all managed care. So the amount of information that comes in and the speed at which it comes in is slowed up. And the notion there is, I think one of the directions that really best has to go, and I think it is going to go that direction, is really to start working more with the claims processor, but at the same time have some kind of relationships with the plans, i.e. in the states in this case, so that in fact you're able to use that data, because that data, that's a population, it's a really important population. And we have a long ways to go to really be able to integrate that population. Now, you should note, it is the case that data sets like United they do Medicare, they do managed care for those. And
Medicaid managed care, yeah.
And there is sort of that. But in terms of, that's usually gets you a plan out of a whole population. And so to really look at a state or look at a region with regard to Medicaid, there's a lot of effort that needs to be put into that. But there's progress being made in that direction.
And in terms of linking in additional types of data, we had some discussion yesterday and earlier today about where electronic medical record information was really helpful on things for, maybe not for some of the key questions related to vaccines right now, but for things like, people need supplemental oxygen, and if so, what type, if that's an important degree of outcome complication that you're trying to track. Any comments or thoughts about the role of electronic medical records augmenting this? I know some of the data sources involved are increasingly rich, including Optum, MarketScan, et cetera, are increasingly rich in those dimensions too.
I'll just mention that for diagnosing bias, we found that reviewing the chart to figure out what really happened under the covers was very useful. So determining that the reason that they came in late was because they thought they had a vaccination side effect and things like that, we actually got from the notes.
And George, your ability to do that, that's not even across different data sources. I mean, Odyssey's a huge global network now. Any lessons learned about how to do that most effectively?
We have a federated data model. You need a strong common data model to achieve it. It's a heterogeneous data model. So we have some sources that are EHRs, like Columbia, where we had the registry link. The registry links came ahead of time. That's why it worked for COVID. And we had the deep information from the notes. But we also use claims data, and we also use international data. International data help because it's larger than the US, and in some cases came earlier. Our Korea database gave us the earliest information about COVID.
So this is Rich. I'll just mention that we do have a large number of EHRs that are included in the BEST system. And we do have some experience with using those. For example, in a recent study that we were doing in Texas, we use the EHR for things such as oxygenation data and mortality data that would not have been easily available in claims. It's HRs to supplement the claims data, but it is a resource that we're building into.
Rich, so I think we lost Rich for a minute there. I think the point was it's beginning to be used more widely. John, looks like you might have a comment on this.
Sure. I mentioned in my presentation that Optum has, in addition to the claims database, a large electronic medical record system, and we have used it. It's part of the what we do for BEST. It's not yet as it hasn't been used for the kinds of analyses that we talked about, the rapid cycle, but it is is sort of there, and it has been used for a number of things, such as investigation of the TTS.
Great, and it sounds like getting there. One other critical data dimension we talked about in the last session and that you all brought up as well is linkage of public health data on vaccination status with a lot of people, and saw the picture of Fenway Park. Glad that Fenway is back to baseball games after that, but the data linkage problems complicating analyses. Any comments there too about the direction we're headed? I frankly heard a lot of encouraging statements in the last session and this one about how much COVID has had a silver lining around facilitating, encouraging more ties between states and their immunization registries and health plans or other data analysis systems to do this important kind of public health work.
Yeah, I agree.
And Rich, good to have you back.
Yeah, just a quick about some of the things that we're doing at Optum, and it's facilitated by our also being a health insurer and a health company that provides health services, and so that some of those connections then to the states to facilitate the transfer of data have happened more rapidly than I was expecting, and they're really helpful for the research. And I think we've achieved a critical mass now, but we want to keep going until we have most of our database covered by that linkage.
Yes, thanks to the hard work of all of our data partners, we do have a number of IIS linkages in place now. I think we're up to 9 states where we're actively exchanging data, and about 20 that we're in various stages of negotiation, and that's helping tremendously. I do think as we look to the future, we really need this to be more complete and faster than it's been. So I think that's an area of making sure that we've got proper data on vaccine exposure for both routine vaccinations, and should any future pandemic happen, I think we've learned that we really need to make sure that we have a way to accurately identify everyone's vaccination status for the purposes of monitoring the safety and effectiveness of the vaccines. So I do think that's an area where we've made progress, but there's more work to be done.
It really does seem like it too, Rich, where if there are some best practices emerging for how to do this, it'd be great to give the remaining states some help and maybe some focused technical support to get over the various obstacles, whether they're regulatory, data sharing restrictions, technical, or just bandwidth. But it seems like a lot to build on here. Sorry, it was George. Was that a comment coming from you, or?
That's Bradley. I was going to just jump in and say that the implications of this, have really been I think highlighted by the current pandemic situation that we're in. But if we look, you were talking about Medicaid a little bit previously, and research on children's vaccines has long been hampered by the fact that vaccine claims in like vaccines for children states using those programs just doesn't appear in claims for the most part. And so really the implications for this across routine vaccinations and particularly in children are pretty enormous. And so this is really great news looking forward.
Yeah, really important public health evidence step for the future, way beyond COVID. So we've talked about data.
Mark, before we leave this, I just want to share one other point that we've heard from some of the states that we're working with. They've experienced situations in which during a crisis like this, they suddenly get additional resources coming their way, but once it's over, those resources shrink and it becomes difficult for them to maintain some of the capabilities they developed. So I do think that's another aspect that we need to consider, how this can be established as something that is regularly available, and we don't have the feast and famine that we're hearing some of the states describe over.
No, I think it's a really important point, Rich. And one of the points of this meeting was to highlight not only the progress that's been made, but especially what the implications are for the future of safety and effectiveness evidence from the real world on vaccines and many other medical products. But vaccines are so important, and so many questions about effectiveness, concerns about hesitancy. This does seem like a solvable problem with a clear and effective policy response that you all shown the way to begin to really address systematically. I did want to shift gears for a few minutes to talk about methods. So we've talked about a lot of steps related to data. In terms of methods, obviously you all put in place some rapid cycle analyses that seems like doing a pretty comprehensive job of addressing whether there are differeses in background adverse event rates versus adverse event rates in individuals who have been vaccinated for now a wide array of adverse events of special interest. And that's not to say it's easy. We talked about the selection bias issues and the importance of having a well-defined cohort. Thank you, George, for the comparisons there. Is that work kind of where it needs to be? And if we're doing so well at adverse event monitoring, rare adverse event monitoring, anything else we need to do? It seems like maybe bringing in more data sources would help, since some of these events are rare, especially if they're focused in specific population subgroups like younger men. Is anything else we need to do in terms of coordination with the spontaneous adverse event reporting systems? Are those still helpful from your standpoint? Just would appreciate some comments about that, whether we have it down yet on monitoring serious adverse events or potentially serious rare adverse events.
Actually, Mark, this is Tom. One of the biggest challenges that we face is really more of a statistics challenge in the following way. I mean, FDA and everybody wants to monitor a lot of events often.
Frequently, yeah.
That is a problem by itself, just in the math. So it's not even so much of a data problem. And we need to do further development of hypothesis testing when we have multiple outcomes. Essentially, we tend to do an outcome at a time, which we know is problematic. And so we get false signals, and then there's panic, et cetera. But so part of this is a way to maybe construct indices or a sum type, so you could monitor in a way where you really can use the power of a test and use the data you can and do it more often. And then recognize at that point, you're really going to be doing kind of more of a investigative report on something to find out, is it real there and what's going on? Because the formal kind of testing hypothesis is fairly limited as far as we can go. And it's not a methods problem. It's a math problem.
Yeah.
Yeah, and just to build on what Tom was saying, first of all, we did spend a decade developing these methods, especially in the Medicare group, looking at safety of flu vaccines. So we had a lot of experience there. But in the current setting, we're running 90 statistical tests a week with the different outcomes that we have and the different age stratifications that we're looking at. So that's just a huge number of tests to be run. And we've done our best to calibrate them so that we'll be sensitive enough to detect an increase in risk, but not have a lot of false positives. But it is challenging to get that right. Just briefly, I want to comment on your question about the passive surveillance systems. Those are still incredibly important. They have already identified important issues such as thrombosis and thrombocytopenia syndrome. They have their limitations. The active surveillance has its limitations, but as part of a system, the passive surveillance systems are incredibly important. Over.
And it sounds like they're remaining so, just a more comprehensive partnership with these more reliable methods for determining differences in population rates and the like. Okay, I'll take silence. Go ahead.
Obviously, I would say, like, we knew a lot coming into it. We learned a lot during it, but there's obviously a lot more to do. Tom mentioned, we're working further. That's one of the projects that BEST is working on explicitly. A big, like, why are we scrambling to get background rates? That's something we need to be doing in parallel and should be ready to compare to anything that comes in. So with modern computing power and with our modern methods, we can at least generate the historical rates very broadly, very comprehensively, being prepared for the next time we need them, including now.
It's a great point. There was a question in the chat too about whether the background rates that you're seeing, George, and some of the Odyssey work are similar to those coming from other sources. And it does seem like continuing to do this work as a baseline exercise, get the results out, get to more comparability is an important, very important step for preparedness. I know we're running slightly, or going to run slightly over as of right this minute, but I do want to turn to the other key methods area. So not just adverse event monitoring, but effectiveness. So we've talked about some of the challenges in developing and the need for real world evidence around effects of vaccines over time, and need for boosters, around potential differential responsiveness in particular groups or to different variants. And that does seem like an area that's sort of more challenging in some ways from a method standpoint, even than what we've talked about. And that seemed like an area we're still kind of working to get in front. I think, Bradley, you talked a bit about some initial work on effects over time to address questions about boosters, but this is a fast-moving pandemic, and looking ahead, certainly going to be some questions about safety, effectiveness, and impact in younger children and in other populations that have been relatively less studied. But tell me how you all are thinking ahead about maybe this. I could start with Bradley, but appreciate others' viewpoints around the methods and the data readiness for some of the questions that may be coming. Some important questions that may be coming about variants, about durability of boosters, about longer-term effects of longer-term benefits of and maintenance of effectiveness of vaccines.
Thanks for that. And I think that's you illustrate a lot of very good points about it. And I think one of the key issues is, often as soon as we first heard what COVID was, I think most groups around the world started putting plans, in place to planning how they would evaluate it. But in many cases, we were writing template protocols or getting ready before vaccines were approved, before we knew how they were going to be distributed, how they were going to be used. And so I think that effectiveness in many ways is really highly dependent on who you are comparing to whom. We've seen some of that data in the around the safety side also, but I think it's really critical, and they're very, very well described and well understood biases, mainly from influenza literature, looking at the really massive differences between people who choose to be vaccinated and people who don't. And then we were all very concerned when we had this very tight, kind of very gradual priority populations, that were rolled out, and that the people who were getting vaccinated were by definition fundamentally different than those who weren't. And so there are, I think, principles. There are good pharmacoepidemiology practices and sound, valid designs that we have to embrace. But in many cases, those just, we had to see how they were going to be used before we knew how we could, who we could best compare to who. And I think it does have to be an iterative process. We've really advocated strongly for the use of negative controls to demonstrate that we're accounting for these some of these biases, and there's a textbook worth of things about it, but I'll let others comment. But I think that it has been difficult. There are a lot of different moving targets in getting it right, but the methods definitely matter. And I think that's one thing. We didn't want to be fastest, but wrong. I mean, I think we want to ensure that we're doing it correct.
Actually, Mark, I think it's worthwhile to mention that CBER has been working on this issue forever in the area of the flu vaccines, because as you know, every year they have the flu vaccines and they have committees that get together that try to then, given the evidence they have, to try to make adjustments accordingly to what the new vaccine is going to look like. So there's a model to plug into there, and I would anticipate this to the extent there are variants and this continues, that would be the framework they would plug into. One of the challenges, as you well know, that has been with regard to doing effectiveness is to get information on who's vaccinated. And the breakdown in the trust between the state and the federal governments has been a huge problem in that regard. And that's got to be somewhat repaired. I think it can be over time, but not in the immediate future, because some of the states don't want to cooperate with the federal government in kind of any way of giving their information, and that's problematic. So once we get through that, though, and we get back on track where it's not so political, I would suspect that any kind of pandemic that kind of comes up and it variants, the FDA does have a model they can go to on that, and I think that's kind of moving in that direction. I mean, FDA's changed a lot in this regard because they're really doing this. The vaccine this time really forced them to do much more post-market surveillance than they had before, and that capacity was there, and that's what's done. But it's not different than what they did with the flu vaccine already.
Yeah, and even some progress on some of the data, the state data linkages. Though you're right, Tom, it's far from universal. Other final comments about effectiveness issues looking forward before we wrap up the session?
Mark, I would just add to what Bradley and Tom said that also the incidence of COVID-19 is a challenge in estimating the effectiveness. Because it's really changed over time in terms of the location and intensity of the pandemic. And that's something that we need to carefully account for in a really high resolution geographic way as we move forward with these models. Over.
Yeah, and again, this is a chance for final wrap-up comments from everyone, but I would just, building on these last few comments, say that one thing I've taken away from this effort is that there was a tremendous amount of capacity for learning about these questions on adverse events, on issues related to effectiveness, thanks to the foundation have been laid and thanks to a rapid progress. Rich, you mentioned at the outset the hard work that all of these data partners have been called on to do in a rapidly developing and rapidly changing pandemic. If there's a key lesson coming out of this, I think that it's a foundation that has many applications going forward, including preparedness and monitoring for the remainder of what's ahead for us with COVID-19, as well as future preparedness for public health emergencies, but also a much better infrastructure for understanding the safety, effectiveness, and effective use of vaccines, and by extension to other medical products. Differences across data sets to make them more comparable and common data models is has shown a lot of progression here in the Odyssey model, and opportunities to leverage some of the questions dealt with from what's learned in being learned in the Sentinel model as well. We've talked about that earlier today too, and a lot of work on methods. The data gets better. It's easier to do compelling work on methods, both because you better understand what you're dealing with and because more confidence that the results really are going to matter. So a great foundation to build on, as several of you emphasized, important that the wind not go out of the sails as we start moving beyond COVID. And it does seem like time to, even as busy as we are with COVID response, time to think about the implications of this whole experience for Sentinel for the future, for BEST, for the NEST program that we talked about as well during this meeting, and for other aspects of medical product safety and effectiveness, real world evaluation. This infrastructure is the most extensive one out there, certainly in the United States, for driving towards more consistency and data science around the data models, around the methods, appropriate methods, and the like. And I think while the pandemic's been very challenging, we've seen a lot of effective responses, and again, a lot to build on. Any final thoughts before we wrap up this session?
I just want to once again thank all of the data partners that have been working with us. We really need this close collaboration for this to work, and there have been times when we've asked for really extraordinary effort from our data partners during this time, and we really appreciate how they've helped us. Thank you.
Yeah, thanks to all of you for the work on this panel and for this critical work on response to this unprecedented public health emergency. And just would also like to thank you on top of that. This goes for all of our presenters, not just this session, for taking the time and effort to share what you've learned to help us build a better common understanding of the opportunities and the challenges and how we're going to make progress with the Sentinel initiative over time. It certainly has been a very important year for finding both important limitations in our capacity to answer key evidence questions, but taking some unprecedented steps to address them. I am not, in the interest of time, going to go back and try to do a full summary of everything that we've heard today. But one of the things that I did want to keep coming back to is this notion of trajectory. Tom, I remember the first time that there was Medicare data run at Acumen. It's come a very long way since then. I remember hiring Jeff Kelman over at CMS and suggesting that he work with FDA on some data sharing activities. Those are small but important steps, and just how tremendous the progress has been over the last couple of decades in getting to better safety and effectiveness evidence with Sentinel and these Sentinel-related programs, NEST and BEST, playing a huge role. I think a big challenge going forward is to build on what we've learned and not lose the momentum that might come as we move beyond the public health emergency. The importance and the opportunities for developing this kind of real-world evidence have never been greater, and the interests and the foundations that Sentinel has helped provide are also broader than ever before. So I hope you all have enjoyed the discussions over the last couple of days as much as I have. I hope you're ready to continue work on the challenges ahead, both in responding to the pandemic and in the broader range of issues related to safety and effective use of medical products that Sentinel was designed to address and is evolving to address, along with the data and the methods and the expertise and knowledge of all of us, on all of you who have been involved in these efforts. I hope we can continue to share these experiences across programs, to recognize that learnings from BEST can be helpful for NEST and can be helpful for other Sentinel programs, as well as these efforts continue to grow importance, which I think they will in the months and years ahead. So looking forward to the next year in Sentinel. I hope it's one that's better from a public health emergency standpoint. Expect it will be, thanks to a lot of the evidence that Sentinel has helped contribute to developing. Before I go, I want to thank everyone who is involved in putting together this very important, timely, and unique event. First, all of the speakers and panelists who shared their work with us, their insights, and not only about what they're doing now, but about how to overcome these challenges and scaling and increasing the scope and impact and effectiveness of Sentinel programs. I certainly enjoyed hearing about all these updates, and I hope you all did as well. I want to give a special thanks to our colleagues on the FDA planning team for putting the time and effort into this, on top of what has been an extraordinary extraordinary year and a half already, including Bob Ball, Steve Anderson, Rich Forshee, Hamila Mouido, and many more. And of course, want to thank our team at Duke Margolis, Trevin Locke, Thomas Rhodes, Aaron Soul, Rachel Hendricks Stirrup, Luca DeRocher, Matt D'Ambrosio, and Brian Cantor. And a final thanks to all of you who attended. Appreciate your being here and appreciate your continued interest and contributions to the important public health goals of the Sentinel Initiative. Take care and enjoy the rest of the day.